Recording in progress.
Ladies and gentlemen, welcome back to the Day 2 event of the Global IR at JPM. My name is Jury Huang, the Head of International Affairs and Public Relations Division at Korea Biotechnology Industry Organization. Yesterday's program was a great hit and had more than 1,500 viewers, it seems. This explains how significant cross-border investment is to the biotech startups today. Cross-border collaboration is recently one of the top interests among biotech companies as the industry matures. That being the case, we would like to start our event by sharing you the recent exclusive rights deal that has been signed recently between Swedish biotech Lundbeck and Korean biotech APRIL Bio. I will now hand the session over to the session chair of the case study, Josh Hofheimer, co-head of Global Life Sciences Practice at Sidley Austin. Josh, we can't wait to hear the story.
Good morning, good afternoon, good evening. Thank you all and welcome back to this next session of the Global IR panel, part of KoreaBIO and BioCentury. My name is Joshua Hofheimer from Sidley Austin, and I'm pleased to be interviewing my two colleagues who've joined me here for this panel, which is going to be a case study on a recent cross-border licensing and collaboration agreement that was entered into between KoreaBIO, a South Korean company, a biotech company, and Lundbeck. And I have with me two of the deal principals from both parties on this session. Sidley had the good fortune of representing KoreaBIO as outside counsel in this matter. And I'm very pleased to be able to have been working with Jamie and the others. I'm going to let everyone introduce themselves briefly. I'll just give you a brief overview of Sidley Austin. We are a roughly 2,000 person law firm with almost, I think, 20 offices globally and 6 in the Asia Pacific region. We have a global life sciences practice that spans the globe and covers everything from research and development and licensing, which is what this is focused on, this session, through to development, commercialization, regulatory, healthcare, GMP, litigation, and the like. Our focus on Korea has been one that we have been pursuing for some time, and I've had the good fortune myself and with several of my colleagues to work for several Korean life sciences companies in their licensing and partnering agreements with global partners and also with regional partners, and as well with some companies looking to do transactions with Korean companies. But that's enough about me. I know you really want to hear from Jamie and Gregor. So let me turn it over to Jamie for a brief introduction of himself, and then Gregor, and we'll jump into our discussion.
Hey, thank you, Josh. First of all, it's very honored to invite this valuable chat, and I'm very glad to share the transaction between Lundbeck and KoreaBIO. My name is Jamie Lee, the Chief Development Officer and the head of the Product Development Division, overseeing the company's partnering activities worldwide, as well as the clinical and regulatory activities. Before joining KoreaBIO in 2020, I was a Chief Business Officer at Curacao, where I was responsible for business development and licensing. Prior to Curacao, I was a Senior Director and Global Head of Regulatory Affairs and the Product Manager at Oxygen, the joint venture between AstraZeneca and Samsung Biologics for development of biosimilar of rituximab. From 2002 to 2013, I served as a General Manager of Business Development and Clinical Development at ESO and led the group that was responsible for business development activities, our latest stage inbound and outbound partnering activities globally, as well as the responsible for clinical and regulatory activities in shaping regulatory environments, accelerating registration in Korea and global territories. I was educated at the Chungnam National University in Korea, holds in Master of Science in Pharmacy, and am also a pharmacist. That's all. Thank you.
Thank you very much, Jamie. Gregor, please.
Well, thank you, Josh, for the introduction, and good morning, good afternoon, good evening, everybody. I'm very pleased to be joining this panel and this discussion. So I'm Gregor MacDonald. I lead the search and assessment team within corporate business development and strategy within Lundbeck. So my team and I are responsible for really sourcing globally assets, research programs, novel technologies that fit with Lundbeck's strategy, assessing these from a scientific perspective, and in supporting their progression through business case development and ultimately deal execution. I joined Lundbeck in the middle of last year after a 25-year career in other big pharma companies, principally GSK and Johnson & Johnson, in business development and R&D roles. So very pleased to be part of this discussion.
Excellent. Well, let's jump right in. So first question, and I'll throw this one out to you, Jamie. How did you get connected with Lundbeck?
Okay, so we are searching the partners by the lead company at the homepage and the partnering conference or our own network, and also, but we don't hire the partnering agent or transaction firm. Okay, so we first met Lundbeck during the US BIO meeting in 2020. So we request a meeting to Lundbeck among the 26 meetings. So during the first meeting with Lundbeck, we found that CD40 ligand, which is our target molecule, is one of the interesting target for Lundbeck. So we are very pleased to, at the first time, to meet Lundbeck in 2020 during the US BIO conference.
And Gregor, what about, were you part of those initial meetings with KoreaBIO, or were you connected with them after an initial gating process? And what made KoreaBIO a potential and an attractive potential partner for you all?
Yeah, so the initial contact between Lundbeck and KoreaBIO actually preceded me joining the company. So these early interactions were through a member of my team. So I got involved in the discussion sort of later in the diligence process. I mean, really, the principal driver of interest for us in connecting with KoreaBIO was the science and the molecular target. The CD40 and CD40 ligand interaction and the resulting signaling pathway that's stimulated as part of the immune response was an area of biology that was highly interesting for our internal research group. We were really looking for a compound to neutralize the CD40 ligand, and we wanted to pursue that area of science through external partnering. KoreaBIO had generated at that time a very strong preclinical data package with their lead compound APB-A1, including data in non-human primates, which we found sort of highly interesting and really drove our overall excitement in the project. And they'd also been able to advance APB-A1 to a stage where it was sort of ready for phase 1 development. So having an asset at this stage of development, firstly, allowed us to really significantly accelerate our entry into this whole neuroimmunology space, which was is an emerging and an exciting area of neuroscience, and really allowed us to start exploring the full potential of this mechanism of action in the clinic. I'd also highlight that KoreaBIO has also some very interesting platform technologies, including their sort of anti-serum albumin Fab associated technology, their SAFA platform. And this essentially allows you to increase the half-life of protein-like drugs and improve their PK profile. So we found this technology platform really very interesting, very relevant to this target, and so we're really curious to discuss further around that opportunity.
Excellent. And I guess on the flip side, Jamie, Gregor has shared what some of their interests were in you all, and the strength of your data, I think, is obviously a key driver in some of the things that you had done early on. But you had 26 meetings, as you said initially, in the US and in Europe. What do you think helped Lundbeck emerge for you all as a primary option?
Okay, so it's a good question. So when we searching the partners, we have three categories, such as 3C. One is the credit, second is the capability, last one is the common history and the continuous growth. Second, capability. Lundbeck has a big progress in the research, development, and the commercialization in worldwide, especially in US and EU. Last one is commitment. We found that Lundbeck has a big, a strong interest and commitment on APB-A1, our lead program, with very swift communication and a very collaborative attitude. So we found that Lundbeck is one of the most great partner for our APB-A1. Lundbeck has focused on the research in the 4 biological clusters, including neuroinflammation and neuroimmunology, targeting the brain function through the innate and adaptive immune system, which is very fit with the APB-A1 program. So this is why we consider Lundbeck as our very good, attractive partners.
Yeah, so you saw opportunities not just for, for this therapy, but for other partnerships potentially, if this succeeded or if the results were positive and the relationship was positive. I guess you were saying you saw opportunity for other partnerships and collaborations with Lundbeck here. That's important. And look, I know also it was one of the things, we had some discussions early on because you all were also considering a regional partner, and doing an initial regional license, and then perhaps doing a global license. But you ultimately chose to go straight to a global partnership with Lundbeck. Can you share a little bit about sort of what drove you in that direction at the end? Because I've advised companies, coming out of Korea that have done several regional licenses with China or other Korean, other Asian, nations, or have done perhaps an EU or US only license initially before going to a full global arrangement. But you all chose a different path here. Maybe share a little of your thinking on that.
Actually, when we set up the partnering strategy, we want to our partner who is responsible for worldwide, not the regional, because our company is very focused on the research initial stage. So we want our partner to cover the globally in the development, including clinical development and also commercialization. We can have several partners in regional, such as Japan, China, EU, and US, but it is very hard to control all partners in each region. So this is why we want to have a partner who can cover the globally, not the regional.
Yeah, absolutely. It's an important point. And Gregor, coming back to you, let me ask you, was this your first, you individually, your first transaction with a Korean partner, or have you had experience engaging and partnering with South Korean life sciences companies in the past? And do you know if this was Lundbeck's first foray as far as a partnership and collaboration with a South Korean company?
Personally, this was my first interaction with a South Korean company. And so, I hadn't had previous experience, although I have, over the last few years been really impressed with the quality of science and, the level of innovation that's coming from the Korean sort of ecosystem. We see now a lot more companies at partnering events from South Korea and really presenting some impressive science. So, this was certainly my first experience of a partnership with a company from this region, but I expect it will be the first of many in the future.
Yeah, that's good. That's excellent. And is this? You mentioned in the beginning that this was an interesting, the neuroscience and neurodiseases are an interesting and emerging area for you all. Is this is the AprilBio transaction something that does fit into sort of an emerging medium or long-term strategy for Lundbeck? And is it something that you see as a likely area that you're going to develop through partnerships, or are you marrying it also with internal development?
Yeah, so I mean, from a Lundbeck perspective, this collaboration fits very well into our overall strategy, which is really moving away a little bit from our historic focus in broader indications like depression and schizophrenia and more into indications that we term niche or rare neurology or psychiatry. And these are indications that are principally treated by specialists, be it neurologists or psychiatrists, and where patient populations are generally more homogeneous and the disease is driven by more understandable biology and underlying disease pathophysiology. I mean, these are often some of the most devastating CNS diseases with very few, if any, treatment opportunities for patients. So, that's essentially our overarching focus going forward. In addition, over the last sort of 1 to 2 years, we've been redirecting our internal early R&D work to focus more around 4 new strategic biology areas, one of which is this emerging area of neuroimmunology, or how we harness the immune system to fight many neurodegenerative diseases. And so, this CD40 biology and APB-A1, for us is really a highly interesting mechanism that we think plays a central role in many neuroimmune indications, and which, for us will offer the potential to develop, new, potent and differentiated therapies for many diseases that fit within our strategy that are driven in part by a neuroimmune response. So, this will be a very important asset in a growing portfolio for us in this sort of exciting area. And I think, it's our expectation that this collaboration will, not only expand our pipeline and accelerate Lundbeck's experience and expertise in this field, but it will subsequently be an anchor asset that will allow us to build out a much broader pipeline within, our areas of strategic focus.
I'm sure Jamie loves to hear that. Jamie, you actually preceded Gregor on this transaction. So tell me, what was the total time from when you first started the process to try to partner this asset, and when and then also when your first meeting was to getting to a signed agreement? How long did it take you?
As I mentioned, we met 2020 in June, and our final signing the agreement is October 2021. So it took around 16 months from the first contact and to sign the agreement. Yeah. It's a little bit long period, but considering during the 16 months, we have a 5 months pause because the first DD started September 2020, but we don't have much data to share to Lundbeck. So 5 months pause for first DD, and we resumed our second DD in February 2021. So accepting the 5 months DD period, and we have also 1 month summer vacation. So we think that around less than 1 year to sign the agreement from the beginning. So I believe that less than 1 month is very fast and good time period from the beginning to the signing the agreement.
Yeah, I mean, I know we first got involved in about I think it was May of 2021.
Yeah, right.
When you all had made progress on the term sheet together. So it was about 5 months, 6 months for our, from term sheet through to definitive as well. So it was in some ways, it was a pretty rapid process, and one that went, fairly smoothly, I think, all things considered. Were there any challenges or surprises from either of you all along the way that, obviously respecting confidences, but that you're able to share from your experience, things that were unexpected for you in this process that you can talk about?
Yeah, maybe I can take that. I mean, there was a few, and to be honest, that's normal for any process like this. As Jamie alluded to, when we started our first round of diligence, that it became clear that there was an important pharmacological study that had not yet been finalized, and not having access to that data really precluded us from being able to take a firm conclusion on the opportunity. We were able to, essentially resolve that, as again Jamie alluded to, by essentially pausing the assessment for a few months, allowing KoreaBIO to complete the study and analyze the data. And actually, this turned out to be very fruitful. It strengthened the data package and really gave us added confidence to proceed to execute the deal. Maybe another thing I'd highlight is that later in the process, we encountered some additional issues around CMC that took us a few extra rounds of discussions, both with the KoreaBIO team and internally at Lundbeck. And here it was particularly helpful that KoreaBIO were able to facilitate us having a direct three-way dialogue with relevant external partners that KoreaBIO had been working with, so other subcontractors. And this really allowed us to talk through our concerns, get explanations and clarifications for some of the issues, and allowed us to resolve that quite efficiently.
What about from your perspective, Jamie?
Okay, so as I mentioned, there was a long pause after first DD, September 2020. The first DD has started only 3 months after we first met Lundbeck at US Bio in 2020. At that time, April Bio didn't have much data to share, so the DD was stopped very early. In order to restart this business opportunity, as well as even to prepare phase 1 IND filing, April Bio had diligently prepared more preclinical and CMC data for the next 5 months. Finally, we were able to restart due diligence with Lundbeck in February 2021 and eventually make a deal. Another challenge is we found that during the agreement negotiation, Lundbeck considered IND approval was linked to making a deal. So we need to focus the IND approval to finalize our deal. But anyhow, we finally got the approval from the US FDA as planned in September, and we can finalize our deal in August after IND approval from the US FDA.
You think that, knowing what you know now, do you think that you would have waited until you had done that additional round of studies before you started the process of meeting with potential collaboration partners? Would it have been, do you think your discussions might have been more fruitful or would have gone differently if you had waited before you had those initial discussions?
Okay, after we signed the deal, we think that before starting the searching partner, we think that developing concrete partnership strategy is very important before searching the partner. I was told that in a good deal making, around 90% of the real negotiation happens before you sit down to negotiation. So no our company, ourselves, and the opposite side is very important. So before we starting the deal or the searching partner, we think that what is our deal structure, such as our licensing, or co-development, collaboration, or option deal, something like that. And which partner is our right partner? Big pharma, specialty, biotech, or new company? So when? So IND before IND approval, or which stage is the right time to have a collaboration or our licensing. So I want to focus that developing concrete partnership strategy is very, very important.
Yeah. And I'm sure you all have a similar mindset, Gregor, on the Lundbeck side. You know, does it make a difference from your perspective when you are looking to these, you know, to in-license these assets? Do you have a sweet spot for where you look, you know, whether it's preclinical or post-phase 1 or post-phase 2? You know, is there a certain amount of, I could say, trust that you have in the external data balanced against your need to see the performance of the asset in your own hands and in your own team's, you know, management? How do you sort of find, you know, that sweet spot? And did KoreaBIO hit that for you here, or have you learned some things that you would do differently?
I mean, we're really open to opportunities really at any stage of development. I mean, obviously, the later an asset is, the more data there is, the more de-risking that has been done, the less risk you're taking on. But of course, then that's reflected in the deal terms as well. So we do look at earlier stage opportunities. I mean, when you are assessing a program at that stage, I think you have to recognize that there will be concerns, there will be missing data sets, there will be unknowns. And, you know, it's often tempting just to pull out and to walk away at that stage. But, you know, one of our learnings from this and other, you know, collaborations that we've been involved in is, you know, to be open about your concerns from the beginning and try to find a way to mitigate some of these risks with the partner, try to obtain or generate that missing data. And you know, this was, you know, what we did in the discussions with KoreaBIO and allowing them to finish a key in vivo study. And actually that can be very helpful for the long-term success of the partnership. It allows you to already start to build a relationship and a trust between the two parties. And ultimately, if that process leads to then a collaboration, it often goes much more smoothly and more rapidly once you start, because you have had that interaction and sort of mutual working through of issues throughout the process.
Yeah, I mean, I think you're exactly right. I have found in some of my, you know, client dealings and relationships that those partnerships that are built on sort of a certain amount of transparency and a collaborative effort and often some early stage evaluations, some things that are done sort of at risk between the two parties, really does go a long way towards building credibility and towards building trust and confidence in both sides and in an understanding of the commitment from both sides that ends up making the ultimate sort of material partnership or license agreement go much, much more smoothly. So I think you make a very good point that it's important for people and for the licensors and the developers and the manufacturer partners to try to be as open as possible and forthright about, you know, what the strengths and weaknesses are, what their concerns are. And when they're communicated up front, you know, they can usually, which you all did here, certainly, you can find ways to to solve the problems technically that you need to solve, but then also, you know, from the legal and structural relationship, find a way to get the answers and to get to the result you need to build that partnership.
Mm-hmm.
So kudos to you both. We're almost out of time, so I guess any advice that you would have, you know, or learnings that you've taken from this that you would apply to your next partnership or your next relationship, you know, Lundbeck, from Gregor, from your side, that you would offer as advice and thinkings to other potential partners, and Jamie, from your perspective, learnings from this deal that you would apply to future interactions.
Yeah, I mean, I think for any collaboration process, especially one like this that, you know, spanned across borders and time zones, and, you know, touched on different cultures, really the importance of relationship building is absolutely critical. You know, these processes, you know, on the one hand are designed to, you know, critique the data and do diligence on the scientific rationale and the supporting data package, but it's also about getting to know each other, about building that trust, about being transparent with, you know, what you need out of the process or the eventual deal, and also recognizing what your partner needs as well. You know, we did this discussion and eventual transaction in the middle of the COVID pandemic, which also had a big impact in that all of this was remote, was virtual. We weren't able to have face-to-face interactions, and, you know, you see the impact of that, being able to meet face-to-face, to be able to address issues. Also on the transaction side, you know, when you're coming to try and to close the deal and some of the final negotiations, it's so much easier to do that face-to-face, and obviously we couldn't do that in this case. So I think that would be sort of my biggest takeaway from here, just again re-emphasizing how important relationship building is in these processes.
Thank you. I couldn't agree more. Jamie, anything from you?
Okay, I also totally agree with Greg's comment on the face-to-face meeting is very important between two licensor and the licensee. Even though we haven't met face-to-face during our deal, I totally agree that go and listen and keep talking to partners is very important. And another comment as a licensor, as a developer, during the preparing the data package, identifying differentiation of our candidate from other competitors in aspect of market, scientific data, development strategy, and so on is very, very important point. How we can develop the differentiation to compare with the other competitors. Even though our candidate is a first in class, we need to differentiate from the others. So if we show the differentiation point to licensee, it's very easy to discuss and to conclude the final deal. And another point is that to contact the potential partners, even though the partners' interest area on their website is not very fit your asset on technology. No one knows very currently and exactly what development strategy, markets, and targets they were. So even though they haven't, they reject our candidate, please continuously contact the potential partners, because there are many BD person in the global company. During the BD person, it may be some different view. So the continuous communication with your potential licensee is a key success factor to have a conclusion out there.
I think we're just about out of time. So let me just express my appreciation and gratitude to both of you all for agreeing to join us here in this fireside chat. And thank you as well to KoreaBIO and BioCentury for hosting this event. And again, I very much look forward to seeing the results and the good science and products that come from this collaboration. So thank you all.
Okay, thank you.
Yes.
Thank you for the great talk. Wow, 10 months from initial contact to signing the actual contract. That is very impressive. Thank you for sharing the behind scenes of the successful licensing out deal. I'm sure many biotechs who are seeking for a licensing deal found the session extremely helpful. Thank you again for putting this together, Josh. For our next session, let me introduce Lauren Grahl, partner at Sidley, Austin, who will be the moderator for Global Companies Pitching Session. Lauren.
Hi, thank you, Jury. On behalf of Sidley Austin, I'd like to thank everyone for joining us today. I'm a partner at Sidley Austin in the Technology and IP Transactions Group. It's been my great pleasure to work on several Korea cross-border licensing deals, both for Korean and for international clients. I grew up in Seoul, Korea, actually, for a few years, while my dad was working in the telecommunications industry. So South Korea means a lot to me, both professionally and personally. And it's my honor to be part of this program. We have a great lineup today of presenting companies and both Korean and international investors. Each presentation will be 12 minutes long, followed by investor questions. So let's go ahead and get started. Our first presenting company is Medicive. Medicive works on magnetic blood purification to treat hyperinflammation, sepsis, malaria, and leukemia. Let's welcome CEO Dr. George Frajum to present.
Thank you very much. Thanks very much. Hi there, Lauren. Hi, everyone. I'm not sure I'm on screen yet. Hi, Lauren. I'm not sure if anyone can hear. There we go. Hi there, everyone.
We can see you.
Excellent. Hi, my name is George Frajum. It's a pleasure to be here today. Thank you so much for the opportunity to pitch. I'm very excited to share our work at Medicive. Could I just ask the host to enable participant screen sharing, please, so I can share my slides? While I'm waiting for that to come through, allow me to just introduce myself. I'm joining you from the UK today. And very excited to tell you about Medicive and the work that we're doing. We have a platform technology that enables doctors to remove several components from the bloodstream with complete specificity, with a large number of different clinical applications. I'm still unable to share my screen, host disabled participant screen sharing. So if the host could do that, that would be fantastic. But in the meantime, I will keep going and tell you about us. So we are just entering our first clinical studies, and we're currently doing a fundraise looking for $11 million, £8 million, to support us as we move through pivotal clinical studies and to regulatory approval. So it's a very, very exciting time for us as a company. I'd love to be able to show you some pictures to accompany that. There we go. Wonderful. Thank you very, very much for the organizers. This is the one. So hopefully that's now up on screen. If I could get a thumbs up from Lauren, is that all good? Excellent. Thank you very much. So here I am. So we have what we call a dual platform. And this technology is able to act both as a direct therapy and also as a drug delivery tool. And I'll tell you a bit more about that. I did want to start off just by thanking KoreaBIO and all the organizers today for the opportunity and for the people joining from all around the world. So very excited to be here. So we have, as I mentioned, a platform tech with many applications, and we're just now starting phase 1 clinical studies. And that is for our first product, which targets and removes IL-6 in order to treat patients suffering from IL-6 driven hyperinflammation. There are several different patient segments who can benefit from that technology. There's a total addressable market for this technology of £20 billion globally, and that's just for the first application of our platform. And as I mentioned today, we're looking to raise £8 million or $11 million to support pivotal clinical studies and regulatory approval. Our vision in the long term is to be able to provide clinicians and stakeholders, including pharma companies, with a single platform technology that can serve on 4 verticals. The first 2 and our current focus are direct therapies. So that is removing something from the bloodstream that is the direct cause of a disease, and also enabling therapies. And you'll hear that this is our second product, in which we remove something in the bloodstream that prevents an existing or new therapy from being effective. And we have an AAV gene therapy enabling technology that we'd like to share with you today. The 2 verticals, which are ones for the future, are drug delivery. So our platform can be used to extracorporeally deliver drugs, allowing you to administer them for a very controlled period of time without ever actually injecting them into the patient. And finally, as a diagnostics support tool, we can purify very, very rare components from the bloodstream, such as human cells, to enable novel diagnostics. So how do we do all this? It's with a technology that we call magnetic hemofiltration. It's a platform extracorporeal therapy. What that means is that we continuously circulate a patient's blood outside of their body through what's known as an extracorporeal circuit, similarly to dialysis. Into this extracorporeal circuit, we infuse nano-engineered magnetic therapeutic beads. So these are magnetic particles to which we conjugate specific binding ligands, such as antibodies. They mix with the patient's blood and bind specifically to the clinical target or targets that we are going after. Those beads and all targets bound to them are then captured by our magnetic filtration system, which prevents the beads and the bound targets from returning to the patient. Using this platform, practically anything can be removed from the bloodstream. And we have already validated this for a wide range of targets, including small molecules and cells. The treatment time is about 4 hours, and it integrates with standard existing hospital equipment. So to be a big blood pump, we make components that interface with those and the therapeutic beads that enable us to deliver our therapy. We are approved as a medical device in the EU and a drug device combination as in the US. And you can see from this that we are able to conjugate any drug to the surface of our beads and enable it to be delivered extracorporeally. If the target's in the blood, you can use your drug for a defined period of time without injecting it into the patient. So we do that with our suite of products. The Mediciv beads are our therapeutic agent, and they're different for every clinical target that we deliver, although we maintain the same core structure. The Mediciv magnet and Mediciv filter work in tandem as the filtration element. The magnet is obviously reusable, whereas the filter is a single-use disposable application. So we sell treatment packs for different indications consisting of Mediciv beads and Mediciv filters, operating a razor and blades revenue model. We have a very exciting clinical pipeline. IL-6 hyperinflammation is our frontline product, and that is entering phase 1 clinical studies today. And the fundraising we're performing is to support phase 2A and phase 2B and initial regulatory approval in Europe. Our second target enables AAV gene therapies. AAV gene therapies are a very, very exciting new area, but they face a problem in that many patients have something in their blood that prevents these therapies from reaching their targets and being effective. We are able to remove that thing to provide a window of opportunity for treatment for those patients. And we have a lot of exciting things in the future. On your screen are some of the things that we've already demonstrated in the lab are possible with our technology, including malaria-infected cells, which we're operating as a not-for-profit campaign. As I mentioned, we're looking to raise $11 million, and that, for those who invest, you'll be embarking at one of the most exciting times for this company. Over the next 24 months, we are going to be going through the key initial clinical studies for our IL-6 product, leading to a CE mark by the end of 2023 to have a market-ready product in 2024. We will also have completed our FDA submission. For our AAV therapy, we will have completed preclinical trials and have a product that is ready to go into phase 1 clinic. And this is our first exit opportunity for us as a company. At this point, we will go around one of two strategies, either an exit through an M&A or partnership, or we will look to IP states to raise sufficient funds to commercialize the product ourselves. So what is the benefit of this IL-6 removal technology? So the problem is that patients have an underlying infection and they have an overreaction to that infection, causing an overproduction of IL-6. IL-6 is a signaling molecule of the immune system, so when it is overproduced, it leads to systemic inflammation, tissue damage, multiple organ failure, and ultimately death. This is known as a dysregulated immune response. It's known as a cytokine storm, as the sepsis cascade. All of these terms broadly define that same phenomena. And what we can do is intervene at that key moment of hyper IL-6 production, providing a rapid and specific reduction in that key inflammatory cytokine that is driving the condition, to bring levels down and rebalance the immune system, enabling the patient to recover. This is fundamentally different to what is possible today with IL-6 inhibitors such as tocilizumab and sarilumab. And clinical trials have repeatedly demonstrated that these IL-6 antibody therapies, when injected into our target patient population of acute hyperinflammation, have short-term benefits, but long-term downsides. And the long-term downsides are caused by immunosuppressive effects that can last for 2 or more weeks. So what we can do is essentially extracorporeally deliver these therapies, providing the short-term benefits, but avoiding the long-term downsides. And that's why we call this a drug delivery. And it works extremely well. We have well over 100 data points demonstrating removal of IL-6 from human plasma and blood. It works at elevated levels of IL-6, such as that graph on the left, and at lower levels of IL-6, such as the graph on the right. On the right, by the way, representative of an average critical COVID patient. Hyperinflammation is a severe driver of mortality in COVID. These are full clinical condition experiments. The only difference between these and what we would do in a patient is that we're removing blood from a donated sample rather than from a patient. But flow rate, technology, parameters, dosage are all exactly the same. We've also completed a lot of safety validation. We've completed animal trials for our full therapeutic product, including our beads, and we're in the process of validating transferred manufacture into aseptic facilities for the full solution for IL-6. We're going to some very exciting clinical studies, as mentioned before, and I'm happy to provide more details on these to anyone who's interested. We put clinical trial design right at the heart of our corporate strategy because I think it's so important to generate the right data and to investigate the right endpoints. After our CE mark, we will have the option of going into one or several phase 3 clinical trials, segmented patient populations, with a strategy looking to demonstrate clinical benefit in truly homogenized patient groups. We have a very strong regulatory team. One thing I've really learned over my years as doing this is that you shouldn't underestimate the challenges that regulatory will put up for you. We have a clear, validated CE marking strategy, and we're embarking on our FDA strategy this year with our pre-sub meeting, which I mentioned to you. We also have a fully operational ISO 13485 compliant QMS, and we'll be applying for certification this year. There are technologies out there today that already enable doctors to physically remove cytokines and other targets from the bloodstream. Dialysis is the most famous, but there are also columns which do compete with us in terms of the patients they're targeting. The big downside of these existing technologies is that they are non-specific and they are not able to go after the single target that's really causing the problem. Therefore, they throw out the baby with the bathwater. There is another company also developing a similar method to ours. We have much stronger intellectual property to them, and we have a different strategy, which I'd be happy to discuss with any interested investors. On patents, we have 2 fully granted worldwide in all major jurisdictions. A quick word on commercialization. In just the 5 major EU markets plus the US, there are 3 million hospitalized patients with a 30% mortality. So there really is a major market out there for our first product. And this is a very conservative estimate of the revenues that we might be able to get over the first 10 years entering the market. We have an absolutely brilliant, diverse team going after this, diverse in expertise and background, and highly committed to the project at hand. We actually have 14 full-time members of staff at the moment, and we're working with an absolute 5-star clinical team and we have some of the top clinical advisors available to us in Europe. Very quickly on the history of the company, we've been going for about 6.5 years now. We've raised £8 million. Half of that has come from grants and half from equity. We've won lots of awards and had some very exciting international press coverage. Please do go check that out and look at our website and engage with us. I've been George Frodsham, telling you about Medisive and magnetic hemofiltration, a platform technology with a large potential market. Our first product targets and removes IL-6 to treat acute hyperinflammation, and we are looking for $11 million of investment. Thank you so much for your attention today.
Thank you, Dr. Frodsham. I think we want to open it now to questions from our investors. And if any investors could, when you are asking your question, please state your name and your firm before asking the question.
Hello, George. This is Jim from KTV.
Hi.
And yeah, thank you very much for your presentation. It's really new for me, and I do think a very unique approach. But my question is, since you are developing your own system and then also like some disposable tools, right? And I'm wondering how's the cost to when you to give the hospital. So, and how about those margins you have about your system and product?
That's an excellent question. Thank you very much. So one of the advantages of our technology is that it does interface with equipment that hospitals already have available in their ICU. So let's focus then on the individual treatment pack for each patient. We haven't finalized a price for that, and to be honest, it will be dependent on the indication and on the market to make sure that the healthcare economics are aligned with what our reimbursement strategy is going to be. But I can share with you that we'd be looking at margins of over 60% per treatment, and that there are some very rewarding revenues, shall we say, that are available to us in there. There's also great opportunity to expand upon that as we scale up our manufacturing capability. These are things that get a lot cheaper as you do scale up. And fundamentally, these are life-saving interventions. So there is plenty of opportunity for both margin and revenue within that. And I'd really be happy to talk to you about the specifics privately afterwards if you'd like.
Okay, thank you.
Great. We have another question, I believe, from Sumin Shin.
Hello, this is Sumin from Partners Investment. I just wonder, it looks like your product will be applied to the acute inflammation situation. You will probably treat one time, and when you get the CE approval, would it be limited to certain specific indications, or can you cover more broad acute hyperinflammation?
That is what the whole purpose of our clinical trial design is, to enable us to apply this broadly to the different patient populations who could benefit from it. So the way we've designed our phase 1, phase 2A, and phase 2B clinical studies is going to generate the data that proves that IL-6 is removed and proves the clinical benefits that are associated with that in a generic acute hyperinflammation patient through an LPS challenge model. And that's why we can segment afterwards into a phase 3 study in defined patient populations. And that enables us to then go after the different patients who could potentially benefit, be they pancreatitis patients in an ICU, CRS patients on the back of a CAR T-cell therapy, or cardiac surgery patients immediately recovering from surgery, or indeed anything else. And that's fundamentally built into both our clinical trial and regulatory strategy.
Thank you.
Great. I think I saw Derek had a question potentially.
Yeah, this is Derek from Life Capital. I just want to have a quick question on first of all, on the competition side. Can you comment on, since Baxter has another filter set that approved for removing pro-inflammatory cytokines, maybe can you comment on the differences versus, I know there's single cytokine versus a set of different cytokines, but how much clinical differentiated factors that do you think you can achieve? And the secondly is, would the product be able to use as an add-on to the existing dialysis systems? I'm asking because in a lot of hospitals, they already have the dialysis system. Maybe it's easier for them to persuade them to purchase if it can be used on the existing machine versus purchasing a total new set of a device. Yeah, thank you.
Thank you very much, Derek. I'll actually answer the second part of your question first. And yes, this can interface with normal dialysis machines, so CRT or hemophersion, as well as the extracorporeal machines that are standard available in ICU. So we don't make a machine. There's no software in our product. We don't have to plug anything into a wall. There's no electronics. There's nothing. We always interface with what's out there. And in fact, we've already got a partnership with a major Japanese manufacturer who's giving us machines for free to ensure that we have complete compatibility with them. So that's absolutely part of our plan. In terms of Baxter's specific device, you're referring to Oxirius, and similarly to the other competitive technologies that I listed on our table, the problem is that they cannot have specificity within their approach, and therefore they're kind of throwing out the good with the bad, and could never have this approach. Furthermore, that type of technology is limited in terms of its ability to remove its targets quickly and to remove the targets when they're low. And you will have seen perhaps from the clinical data already published from the technologies that are out there that they are really struggling because of these challenges to prove a meaningful clinical benefit. That being said, they're on the market. They have first mover advantage, so we will have to compete with that. But we do believe that what we're offering, we know that what we're offering is very unique, and we believe that that uniqueness is what's going to make the difference clinically. And then behind that, we have a whole platform of opportunities to follow on, and that's something that none of these companies have.
Well, great. Thank you so much for your time. Great presentation. I think due to limited time, though, we're going to move on to the next one.
Okay, thank you so much for your time and attention, everyone. Please do engage with me if you'd like to.
Great. I'd like to introduce Manoj Bhojwani, the CFO of SkinVision. SkinVision focuses on using AI-based smartphone app medical devices for skin cancer detection. Let's welcome Manoj.
Hi, can you unmute yourself?
I hope you can see my screen now.
Thank you.
Do you see my screen, just to check?
Yes, we can see.
Okay, good. Thank you. Thank you very much. And thank you for the introduction. So where I'm here today to present SkinVision. What SkinVision is an app that helps diagnose or assess skin cancer risk. So a bit of a background before I get into this. 1 in 3 people globally are actually impacted by skin issues on a daily basis, of which 1 in 5 actually, unfortunately, get skin cancer in their lives. 1 in 5. These incidences are higher in countries like US, Australia, Europe, but actually it's globally the same, more or less, with 1 in 5 to 1 in 6 people getting skin cancer. What our solution is? Our solution is simple. We have created this app, which is actually available on App Store and Android. It's already commercialized in Europe, Australia, New Zealand. And now we are working through the US FDA. So we already have the US FDA breakthrough recognition and also have the Europe CE mark. It's already commercialized and being sold and operated both in B2B and B2C channels in these markets. What is happening and why this is needed? We have an increased demand for healthcare. Why? Because the incidence of skin cancer are increasing. 5 million in US, and more than 7 million new skin cancers were diagnosed in 2021 across the world. This is more than 40% increase compared to a decade ago, and almost doubled compared to 2 decades ago. This creates a huge restraint and pressure on the healthcare system, because a lot of appointments related to skin cancers are actually not given in time. So the waiting times typically are 3 to 6 months, both in mature markets and emerging markets. And to get to a dermatologist, so pass through the GP and then get to the dermatologist, get the scan, it can take up to 9 months. Unfortunately, it's sad, but 95% of the skin cancers are treatable, curable, provided they're diagnosed in time. But if they're not diagnosed in time, this can be actually quite fatal. And that's why there's a huge unmet medical need, especially in COVID times, which is proven that the digital health solutions like this can work. What do we have now? We already have clinical studies based on which the CE mark has been approved, and it's been commercialized in Europe and Australia. And now we're working through the FDA to move on from the breakthrough to a full US clinical pathway. And with this technology, what we have is we have a machine learning artificial intelligence database, which kind of every time a user takes a picture, the image is then compared to the cloud server, and it gives within few seconds, just a few seconds, it gives the assessment whether this is a high-risk cancer, it's a high risk or a low risk. If it's a high risk, then the patient has to go and see a doctor, and that also creates a rationale for an appointment. If it's a low risk, then there's no need to go to the doctor. So what our mission is to get the right individual to the right doctor at the right time. Because what happens is that if people are going to the doctor when it's not needed, it's creating unnecessary pressure on the healthcare system, whereas people who are needed to go on time are not getting the appointment in time. And therefore, this is also our model from a health economics point of view, that we do not want people to go when it's not needed, and they should go in time for the expediting of the patient journey. The market is huge. We have a roughly $200 billion of skin cancer market or skin health market, and also about $190 billion of the skincare market. And why I mentioned these two markets together, adding up to just under $400 billion addressable market, is because today we are in skin cancer. We have these images clinically secured, and the artificial intelligence machine learning app documented through clinical studies in Europe. In future, we want to go beyond cancer. So we want to go into areas beyond cancer, and that's where the big skin care market will also be untapped. This is an example of the images that the app actually took in Netherlands alone. So our company is headquartered in the Netherlands, and of course we are a global company headquartered in the Netherlands. And this sample shows that in 2021 alone, 2,500 cancers were detected in Netherlands as an example by this app. And these spots are real. These are not animated. These are actually real spots of where these cancers were spotted. One more thing on this slide is that actually, since this is a machine learning and artificial intelligence algorithm, every time a picture is taken, we also have a team of in-house dermatologists who actually go and do a quality control check. And we keep on improving. The machine keeps on learning, and also we keep on improving our sensitivity, which is already at 95% today. I'm going to move so that we can have more time for questions. We have a very credible team here. So Eric, our CEO, myself here, and Francesco, coming from a background of regulatory and operations, and the CTO, Tim, who is actually leading the AI and ML team here. We also have an experienced board, already have venture capitalists. So we have already got seed and A in the past. We are now looking for Series B. We also have a very world-renowned scientific board based on some of them who are also in the studies involved, one from Erasmus here in the Netherlands and another from Munich in Germany. We continue to do these studies in each of our focus markets. In the US, we're going to start soon. We already have these studies happening in Europe, UK, and Australia. Now, what we want to do, we want to raise 25 million euros as part of Series B, and the main reason to raise this is actually to invest in these 4 areas. One, we want to, based on this proven model in Europe and Australia, especially in the Netherlands, where it's already profitable, we want to expand outside. We want to go deeper into markets of Europe, which is not fully penetrated yet. We are expanding there, but also go into Asia Pacific, Latin America, and more importantly, US with the FDA. And there we need also to invest in the US specific studies, and that's where the FDA and the NICE of UK, and that's where we want the investment to be done as well. At the same time, we want to invest in the platform, because the platform is with the innovations coming in AI and machine learning, we keep upgrading our platform. And last but not the least, at the bottom right, you see here is the investment to go beyond cancer. So we want to go into other skin areas with the same proven technology and business model that we have on the cancer assessment. We will do this in the beyond cancer area as well. So with this, I wanted to really go fast, catching up with time, but actually have more time for Q&A. So I decided to do a short presentation so that we could do more of the Q&A.
Great. Thanks for the presentation. I think we actually have quite a few questions. The first one, I believe, request was from Min Shin.
Hi, thank you for the presentation. My name is Min from KB Investment. I just wanted to ask you about some of the general questions, such as how do you actually generate revenue? That was the first question. And second one is that, when you're actually, taking, diagnosing, well, it's not diagnosing, but screening for whether you should visit a doctor or not, it's always come with a liability issue. Would that be any risk for this?
Yeah, good questions. So first one on the revenue, we have 5 types of revenue models, broadly categorized into 2, B2B and B2C. In B2B, what we have in the business to business, we have actually is the contracts with health insurance companies, especially in Europe, where the market is typically reimbursed by the health insurance company. So in Netherlands, for example, more than 40% of the population is already reimbursed based on the health insurance contracts we have today. So what users have to do is download the app, and they simply key in their ID, which then the app recognizes it's paid by a health insurance company, and they access the app for free. Why health insurance companies pay for it is because we have proven to them with health economics assessment that 2 reasons. One, not sending the people who are not needed to go to the doctor, we save time and money there. And second, sending the patients who need to go to doctor fast also helps early diagnosis and therefore saving on the late diagnostic expenses. The second revenue stream is on the business to consumer. So we also have 25% of our sales today coming directly, people paying out of pocket, whether the health insurance reimburses or not. And that's typically coming from outside the Europe, as well as some countries within Europe, where we are not yet having the health insurance contracts like in Southern Europe. So these are the 2 broad categories. We also have 3 others. I can elaborate that in detail maybe in a specific session. The other question you mentioned, we currently do not call ourselves as a diagnostic tool. So that's a very important distinction. Although me personally, I think this is as good as diagnosis. But at this moment, we are not making a claim of diagnosis. What we are calling it is an assessment. And what assessment means is that it is a filter to assess whether the risk is high or low. So the legal liability is not there or minimal, because all we are saying this is high risk or low risk. In reality, the app is capable of the machine is capable of saying which cancer, what stage, but we are not going that at the moment. We are only keeping it to an assessment.
All right, I think maybe one more question from Anai Park.
Okay, so I'm Anai Park from Daily Partners. Well, actually, my question kind of touches on what you have answered previously, but I was kind of curious if our app has been beneficial for early detection in the clinic.
Yes. So the app is definitely helpful in the early detection, because the app has 500,000 images, of which 100,000 images were secured and in a clinical setting. And therefore, each time we feed the app and educate it even more, the machine, it gets more and more samples. So it actually helps with the early detection, because people may not realize that the the molds can actually be dangerous. And the app is capable of saying, okay, this may look like a simple mold, but it is actually high risk. Please go and see a doctor. So it has proven, we have studies which show that it has proven early diagnostics as well as expediting the patient journey and saving lives. We have so far 40,000 to 50,000 early detection of cancers done through our app since inception.
Great. Thank you so much, Manoj. Really enjoyed your presentation. All right, let's
Thank
move
you.
on
Thank
to
you
the
for
next one. Thank you. Our next presentation is by Arcturus, and presenting for Arcturus is Dr. Martin Emanuel Bittner, the co-founder and CEO. Now, Arcturus provides a rapid automated R&D platform for drug discovery. Let's welcome Dr. Martin Bittner.
Wonderful. Thank you very much for the invitation. It's very nice to be here, and I'm very glad to speak to you about the company. First of all, can you see my slides?
Yes.
Wonderful. I can see people nodding. Excellent. In this case, let me give you kind of a brief introduction regarding who we are and what we do, and most importantly, why we do it. First of all, as a company, we believe that right now in biomedical research, we're faced with certain challenges. Above all, data quality in biomedical research right now is oftentimes extremely poor. Many of you will be familiar with the reports that Bayer and Amgen have been working on a few years ago, which have shown very clearly how a majority of drug discovery findings could not be reproduced by third parties. At the very same time, data generated in manual experimentation oftentimes is not generated in a way where it would be fully compliant with best practices of experimental procedures, where it would capture relevant data and metadata, such as temperature, humidity, and where it oftentimes is generated in a way where data density is less than what it could be and what it should be. At the very same time, we do live in an environment where there are numerous pressures on every single biotech and every single pharma company to complete more projects, investigate more directions, and make sure we do select the very best programs to move forward. And finally, our most important resource, our most valuable resource, are our scientists. But right now, they spend most of their time manually pipetting small amounts of liquids, when instead there are so many papers to be read, so many discussions to be had, and so many ideas that would need greater intellectual input than just pipetting small amounts of liquids. That is the reason why Arcturus, as a drug discovery company headquartered in Oxford and Boston, uses an approach which is very heavily enabled by automation. We built a drug discovery company that leverages robotics, automation, data science, machine learning to conduct drug discovery in the best possible way. Many of you are familiar with high throughput screening and traditional automation in drug discovery. We do things very differently. We rely on end-to-end automation of entire research processes, from targets to hits to lead to candidate, where we generate extremely rich, fully reproducible and reliable data in areas such as cell biology, molecular biology, and biochemistry. And we then use that data to make the best possible decisions on which programs to move forward. Because in the end, drug discovery is nothing but a series of decisions. Which target to focus on, which hit to choose, which lead to progress. And our goal is to make all of these decisions based on fully robust, highest quality data. And what that means is we can progress programs with far shorter cycle times. We can run experiments with a far greater degree of efficiency, reliability, and resilience compared to traditional models. And as I mentioned, the technology that we've developed is highly differentiated from what you would have seen in, for example, high throughput screening departments in pharma as of now. We're moving from a single automated process. We've been focusing very heavily on flexibility, modularity, automation from end to end, and a breadth of processes and breadth of experiments, which gives us a huge edge compared to any other company that is working on automation in drug discovery. This slide is a very brief overview just to show how we move from target ID to assay development to hit ID through to lead and candidate selection, where across the entire breadth of the process, we have our own proprietary technologies which emerge at the intersection of robotics and software, which allow us to run experiments in a time frame and with a resulting data quality which is second to none. That means timelines can include setting up entirely new assays in just a few hours, in running entire studies in a few days, and in generating data which has Z primes and standard deviations which go far beyond of what is normally possible in a manually operated environment. And we do have a set of proprietary technologies that emerge, as I mentioned, at this intersection, and that in most cases are already patent protected or filed for patent protection, which allow us to run these experiments in this next generation approach. So to summarize kind of these points and how we do drug discovery differently compared to others, think of the traditional drug discovery process as this zigzag line taking you from hit to candidate, where at every single step, you might want to go left, you might want to go right, there will be mistakes, there will be shortcomings, there will be false negatives and false positives, and the overall process will take longer and will arrive at an inferior outcome compared to what it could be, where in our case, we need far fewer steps to move from one generation of molecule to another, to a superior one, because every single step of the way, every single junction is powered by better and more complete data, which includes complex cell models, spheroids, organoids, primary cells, as well as entire mechanistic biochemistry as it is required for every single decision. And what we've done over the past few years is make sure that the process, the underlying DMTA cycle, is automated to the fullest degree possible. From generating new molecules to analyzing data to running experimentation, with the only missing link being kind of automated chemistry, which is currently under very active investigation. But the overall process runs in an, for the most part, automation and robotics powered way, which gives us this unique edge in data quality and in confidence in our findings. And as I mentioned briefly, in terms of focus areas, we apply our technology across cell biology, biochemistry, molecular biology, where all of these experiments serve a single need, which is allowing us to ask really difficult biological questions for novel target classes in oncology, in neuro, and to identify the most promising targets, hits, and programs to move forward. And this is our focus as a company. So you see kind of the two sides of Arcturus. On the one hand, we have an internal pipeline of drug discovery assets, which we progress either on our own or in partnership and co-development deals, where we progress these programs from novel targets to hit, to lead, and beyond. And at the same time, we also have several partnerships with other biotech companies where we've seen synergies and where we've seen a complementarity in their approach, their knowledge, and our own, as a way to make sure that we progress programs which can really address an unmet clinical need in the best possible way. So you can see Arcturus is a platform company and at the same time a portfolio company where we have an active range of programs reflective of some of the most promising technologies currently out there in small molecules, in PROTACs, in novel target classes, all of which are enabled by the underlying platform technology which we've designed and built over the past few years. In terms of company status and locations, we are headquartered, as I mentioned, in Oxford, as well as in Boston with our leadership team and kind of our robotics and our platform. At the same time, we also maintain a small hub for the Asia Pacific region in Singapore, because we do have very active collaborations in Korea, in China, in Singapore, in Australia. So for us, being international is kind of part of the DNA, so to speak. When it comes to team and background, so kind of there's basically myself, so you can recognize kind of me, plus kind of a team of advisors, board members, C-level, all of whom bring years, in many cases, decades of experience in pharma, in biotech, in robotics, and to help us build the best possible drug discovery company we can. At the moment, we're raising our Series A, and within the Series A, we're going to focus on expanding our robotic platform even further, expanding our team with additional key scientific and commercial hires, especially in our Boston hub, and make sure that our internal drug discovery programs are progressed into IND-enabling studies, ideally up to phase 1. The size of the Series A will be a range of $30 to $40 million, and we're currently speaking with several potential funds for a syndication that will allow us kind of to raise these funds and progress the company in the best possible way forward. And with that, I would like to leave it here, and thank you very much for your attention.
Great. Let's open it up to some questions. I believe that there's a question from Erin No.
Hi, this is Erin No from Intervest. Thank you for your great presentation, and then it was very interesting. You mentioned that Arcturus' system is all fully automated systems, but it must be based on some kind of data sets to analyze. And then I want to know the source of those data. Is it like public, or was it like generated from the pharmas or other sources?
Yeah, so if we look at drug discovery companies right now, most companies work with public data sets or work with external data sets. And that is kind of where part of the problem lies, because you need to have better data to make better decisions. And that is the reason why all of our data is generated in-house. So we built a robotic laboratory, basically a wet lab, but automated, where we generate data in-house, which we then use to drive our decisions in target ID, hit ID, and so on and so forth. So see us as a wet lab company, but robotics instead of people in lab coats.
Great. Thank you so much. I think we're out of time, but a really great presentation. Really enjoyed it.
Thanks very much.
Thank you.
I'm going to share my email kind of in the text box so that people can feel free to reach out anytime.
Perfect.
Thanks.
All right. I think we're going to move on then to our next presentation. Our next presentation is with Blue Jay Therapeutics. And the presenter will be Dr. Keding Chu, founder and CEO. Blue Jay Therapeutics is developing antibody and small molecule therapies for functional cure of chronic HBV infections.
Hello? Hello, can everybody hear me?
Yes, we can hear you.
Okay, great. Yeah, sorry about it. I had an internet glitch. First of all, you guys can see my screen as well, right?
Yes.
Okay, great. I want to thank the meeting organizers, KoreaBIO and BioCentury, for inviting me here to present Blue Jay story. And So here's an overview of Blue Jay. Our mission is to develop innovative cures for infectious diseases. Our first target indication is chronic HBV infection. Our strategic focus is on reduction of surface antigen and restoration of the adaptive immunity of the patient to achieve high cure rate. As I will tell you later, we have 2 lead programs. We licensed exclusively from Novartis when Novartis strategically exited the infectious diseases. Our lead program is BJT778. It's a monoclonal antibody, best in class, with very high potency and very good preclinical data to show the high potential to be a very critical component for functional cure for HBV in the combination therapy. Our BJT574 is an oral available small molecule HBV inhibitor. It's first in class, and this is one of the other components we will have for a high functional cure rate of HBV. Of course, I'll show you, we have a very strong and experienced management team with a track record for successful drug development in antiviral drugs. And people first, here are the people. As you can see, everyone here are very experienced in their own way. And probably the one youngest is Hassan, who only have 1 NDA, and everybody else have multi-NDA approvals and multi-IND submissions. And we all know chronic HBV is a major global healthcare problem. More than 240 million people worldwide are infected by this disease. And more than 850,000 people die from HBV-associated liver diseases. So current standard of care is very good in inhibiting virus replication, but with very low cure rate. Depending the regimen and the compound you are in, it's anywhere between 0 to 11%. And we all know HBV virus is a DNA virus itself. It's not a cytopathic. The problem was really the existence of the HBV cccDNA and the HBV DNA integrated into the human chromosome. Even though the replication is very well inhibited by current standard care, but the virus protein are continuously made, so continuously elicit a antiviral protein immune responses and causes chronic inflammation, liver cirrhosis, and the liver cancer. And here is a kind of a current landscape in the drug development effort. Of course, there's some effort still trying to inhibiting viral replication on capsid inhibitors and next generation nucleoside analogs. But the majority of the effort in the industry are focused on surface antigen reduction. And the biggest effort at this group of companies, J&J, GSK, Roche, Vir, and Arbutus, are trying to use siRNA, antisense, or microRNA to degrade HBV RNA in HBV infected cells to reduce surface antigen. And the second group, but all those clinical data has came out already. I'm sure you follow the field. Most of them, maximum could reduce surface antigen in 2.5 log. On average, it's about 1 log. So that's really not good enough. Nobody were able to clear surface antigen to undetectable level. And the second class of compound people are using called the nucleic acid polymers. So the company Replicor and Oligos all had a compound like this. For unknown mechanism of action, this compound inhibit surface antigen secretion from HBV infected human liver cells. So Oligos data just came out. You probably saw was not, but they stopped it because of the lack of efficacy. And Replicor had fairly convincing clinical data, but it was very hard to verify the clinical trials because the way they conducted the trial. And the third class people are focusing on are some people think there are deficiencies in the innate immune responses in chronic HBV patient. So Gilead and Roche are developing toll-like receptor ligand agonist or RIG1 agonist. And the toll-like receptor 7 and 8 agonist data came out by Gilead already, has very low cure rate, about 3 to 4%, and a significant side effect. And RIG1 data is not out yet. And of course, the antibody program, there are two companies, Vir plus Blue Jay. We have monoclonal antibody trying to use as a very effective agent. I'll show you data both in mouse and in patient to reduce surface antigen from the patient peripheral blood. So this is just an overview of our pipeline, Blue Jay. As I said, we have a best-in-class monoclonal antibody, BJT778, and we have already started GMP manufacturing, CMC, in collaboration with Lonza. So this will be in the clinic end of this year. And we have a small molecule, oral available, first-in-class program as well, 574, which we started the GMP manufacturing with WuXi, and this program is expected to go to the clinic third quarter next year. And we're at the same time making a liver targeting 574 so we can specifically increase the efficacy and reduce any potential side effect of this oral small molecule compound, and this expected to go to the clinic as third quarter of next year as well. So here is, you guys probably already heard from immuno-oncology, PD-1, and a number of others are immune checkpoints. And in chronic HBV, the immune checkpoint is surface antigen. That's why the whole industry is aiming at reducing surface antigen. Same here. Our focus is also on reduction of surface antigen. The high surface antigen viral load really is the one leads to T cell exhaustion, B cell exhaustion, so chronic viral infection. And reduction of this should restore T cells and B cells to their active immune state, can be activated, and to really resolve the virus infection. And to that end, like I introduced before, our BJT778, I will show you data. It will be the most simple and elegant way to clear surface antigen out of peripheral blood. And the BJT574, our oral small molecule, will be able to clear surface antigen in HBV infected liver cells. So BJT778 is a human IgG1 anti-surface antibody, antigen monoclonal antibody, best in class. So I made the statement, antibody is the most simple and elegant way to clear surface antigen in patient peripheral blood. And that's actually clinical data is shown here. This is a clinical trial conducted back in 1999 by a company called XTL Pharma based in Israel. At that time, surface antigen as a checkpoint blocker in chronic HBV infection was not very well established. So they're still trying to reduce viral load with their antibody. This is a nanomolar antibody binder. They also had one problem because in the 90s, people are still not that good making therapeutic antibody yet. So they had a PK problem. They only had 2 days in patient, but nonetheless, even though they failed to reduce viral load in this patient population, they closed the trial, published the data 2002, but they measured surface antigen in those patients. As you can see, this is a phase 1 dose escalation study with 4 different cohorts from 10 milligram to 20 to 80 milligram. Each cohort, there are about 8 to 12 patients. And as you can see, even at the 20 milligram cohort, you already see patient had more than 3 log surface antigen reduction. And this patient surface antigen is completely undetectable. And once you got to 80 milligram cohort, maximum reduction and average reduction is very close. They're all around 3.5 logs. So there are 10 patients in this cohort. None of them surface antigen are detectable in their blood. Just to comparison, to show you the siRNA data, and this is a representative. This is a Abuta's siRNA, which just like any other RNA as well. It can reduce surface antigen maximum 2.5 log, but on average, it's about 1 log. So this result to a immunologist like me is not surprising at all. The reason our adaptive immunity evolved so exquisitely with T cells, B cells to produce antibody, this is really part of the function of an antibody. The antibody's function is to clear bacteria particles, viral particles, and fungus particles. And surface antigen, HBV surface antigen exists in the patient's blood, either as a viral particle or a subviral particle, which is why antibody is the most effective way to remove surface antigen from patient blood. And here's the BJT778. As I said, very potent, a picomolar KD binder, and also picomolar IC50 in neutralizing all different serotypes and genotypes. It's a human IgG1, and it has 20-fold higher affinity binding than the XTL antibody I just showed you. So this is the detailed of the affinity of this antibody. How good is it to remove surface antigen? Here is in the HBV mouse model where antibody was dosed 1 dose, because this is a human IgG1, so it has about 5 days half-life in mouse, and it was able to reduce surface antigen right away more than 3 log. And the dynamics totally correlate with the half-life. And here's the other one, Vir, like I said, also have an antibody. And the dark blue line is the Vir antibody, where bi-weekly dosing for 2 weeks, 4 dose, they were able to only reduce surface antigen by 1 log. And Vir also have an siRNA, like everybody else. siRNA can reduce surface antigen on average about 1 log. The most interesting thing is when they combined their monoclonal antibody with their siRNA, which is the orange line, now they can actually reduce surface antigen more than 3 log as well. And this actually translated quite well into patient. This is Vir's data in patient. They reported December last year in the AALSD meeting. This is also dose escalation with 3 different dosing, 6, 18, and 75. You can see there is actually a very nice dose response trend in surface antigen reduction. And it happens very quickly as well. And the most remarkable thing is really at 29 days, the effect still lasted till then. So we think our antibody, because our higher affinity to the surface antigen, and we are able to reduce surface antigen in the animal models alone with 3 log. So once we go to the patient, we think we're going to be very effective, clean surface antigen to undetectable level in peripheral blood as well.
Thank you, Dr. Chu. I think we want to make sure that we leave at least a couple minutes for questions.
Okay, just 10 seconds for BJT574. So it's a RNA degrader. So as you can see, treatment of the compound can reduce surface antigen to non-detectable in HBV-infected liver cells, which translate into the 1 log reduction into in the peripheral blood, just like siRNA, except we are small molecule are available, and our compound very potent with very good drug-like property, and we are coming out raising 60 million, and trying to complete our phase 1B readout, interim readout, which is exactly this significant value inflection point where Arrowhead siRNA had an interim readout phase 1B. J&J did a deal with them, 3.5 billion. But early on, this end of this year, when we dose our first patient with our antibody, we'll be able to reach this value inflection point, which is Dicerna's siRNA. Those patients, Roche gave them 200 million upfront, 1.47 billion in milestone payments. So I hope if anybody interested can come contact me, and love to tell you more about our program.
Great, thank you. I think let's just have one question here, and I think that Jiwang Chun has a question.
Yeah, this is Jim from KTB. So I have 2 questions. So the first question is, so in terms of your antibody program, so where do you planning to manufacture for your clinical some DP, and also how do you manage the CMC of your antibody program? And second question is about your small molecule asset. That so how is the liver metabolism study, and then want to know the biodistribution. So is it go very well to the liver target? So those 2 questions from my side.
Very good question. So the second question on the small molecule, because we licensed it from Novartis, we have not generated any data ourselves. Novartis did not do a liver distribution, but because Jeff, with 20-some years chemistry, medicinal chemist experience, he actually took a look at the structure, and he predict this actually would have liver accumulation. But we're busy generating those data right now, and we completely fully realize, just like you are now, we want them to be accumulating in the liver, right? Because we want to target the liver. So we just started a very sophisticated liver targeting program, because Jeff's experience, he had been in Gilead, did a lot of the specific liver targeting in Gilead already. So we started that program. So aiming at 3rd quarter next year to go to the clinic with our liver targeting. So our goal is liver versus the peripheral more than 20-1. And regarding the monoclonal antibody, we started our collaboration with Lonza 3rd quarter last year to start GMP manufacturing CMC. I didn't have time to introduce you to my star in the company, with Kevin Lim. Kevin and I were classmates in medical school in China. And Kevin, like me, came to the North America, did his graduate school training. But unlike me, in the past 30 years, he had just focused on one thing, which is CMC, GMP manufacturing. Kevin has done those kind of in Bayer 10 years, and CDMOs with Catalan and mid-sized companies. In his professional life, he has manufactured 22 biologics, 18 of them are monoclonal antibodies, and he submitted 8 INDs and submitted 3 NDAs. So we're really fortunate for Kevin to be the first one join us and managing all those. Yeah, you're right. It's very complicated. We're really happy we got the right person.
Great. Well, thank you so much for your presentation. We appreciate it.
Yeah, thank you. Sorry about the initial glitches.
No worries. All right, so we're going to move on now to Wealthy Therapeutics. And the presenter is Abhishek Shah, the co-founder and CEO. Now, Wealthy Therapeutics is developing software as a medical device using its Wealthy Care digital therapeutics platform.
Thank you so much for having me over. And first, I'd like to thank the organizers for setting this up. My name is Abhishek Shah, and I'm the co-founder and CEO of Wealthy Therapeutics. Just doing a quick screen share. We set out on a mission to be able to impact 10 million lives in this decade. And the way we sought to do that was to imagine a world where every single drug out there, whether in R&D stage today or in commercial, would have an active digital ingredient that pairs up with its active pharmaceutical ingredients so that software as a medical device can work as software-based medicines to complement molecule-based medicines. And the combination really drives unmet patient needs at scale and is able to combat real-world outcomes and drive the promise of healthcare to the next level. So we as an organization, we're exactly that company. We've built out what we call an operating system, a base platform on top of which individual condition-specific software as a medical device solutions have been built out, have been co-developed sometimes with pharma, and have been commercialized or are in different stages of commercialization. We today work with 10 enterprise partners across the globe in Asia and the EU, across cardiology, reproductive health, diabetes, pulmonology, and rare diseases, as we work towards taking these active digital ingredients and driving them to market. We work very specifically across these geos. We've been really fortunate to show through our 85,000 patients that have been prescribed one or more of our software as a medical device solutions. Through these 10 enterprise partners, over 30 real-world publications showcasing what we do and how it works and how it can drive real-world outcomes at scale. We've been able to deploy standalone digital therapeutics, which are right now in clinical trials. Like, for example, we have one in a rare disease undergoing a clinical study in Spain. We have a companion digital therapeutic working in recurring pregnancy loss, partnered with a large pharma company. We have a diagnostic digital therapeutic focusing on finding people who are at risk of a particular condition before funneling them to a medtech partner of ours. We have a combination digital therapeutic, which is packaged with a medtech device and is sold nationally in a country. And we've built revenue centers for a few of our biopharma companies as a result of this as well. The way this works is we have a base platform. That base platform has both the regulatory infrastructure in place as well as the capability infrastructure in place. And on top of this base platform, condition-specific digital medicine solutions are built out. The regulatory infrastructure today allows us almost access to most countries in the EU and a lot of the countries in Asia. It's got the entire QMS capability, Class 1 and Class 2, from a CE mark perspective. And we are right now building our regulatory capability for the US. And by the end of Q3, beginning of Q4, we will have that in place in this year as well. The platform has all the remote patient monitoring, connected device, clinical care coordination, and one-to-one care support where required, and the complete and total capability to design a patient journey, HCP journey, and a companion to the drug journey to be able to enable digital therapeutics to get out there in front of patients. We today, on top of that base platform, have been fortunate to already deploy indication-specific digital health solutions out in the market or at different stages of clinical validation across a few therapeutic areas. As I mentioned earlier, we today are doing feasibility studies now in oncology and immunology, and that goes live again in Q2 and Q3 to shore up the portfolio even more. The way we operate is we come in to identify unmet patient needs, which the drug either was never designed to solve for, or the drug is causing in the first place. It could be symptoms, quality of life, quality adjusted life year related. And then from those unmet needs, we identify the meaningful aspects of health that matter, which are not being solved by the drug. We identify the clinical and non-clinical endpoints that need to be given or need to be influenced clinically significantly. And then we go out and design a digital medicine solution, which is attempting to solve just that. We specifically work with our pharma partners at very specific stages of their molecular journey. When we work with partners at a phase 2 or a phase 3 level, it's much more about how do we use remote patient monitoring and how do we use patient biomarkers or discover patient biomarkers to assist in the phase 2 or phase 3. And then once a patient gets out of phase 2, then how do we get them on real world evidence generation? When it comes to the rest of it, especially when molecules are going into launch, growth, and also approaching LOE, how do we work with them to be able to build either companion digital therapeutics, which have standalone claims but work really well with the drug, or combination digital therapeutics that work together with the drug to build joint claims, which can be stronger than the drug could give standalone. And across the board, we're able to build remote patient monitoring solutions, of course. And in a lot of cases, we're able to take archaic patient support programs and convert them into real-world evidence generation machines, which then in turn build the pathway towards companion or combination digital therapeutics. We've been able to work with our partners now in very specific use cases to drive business ROI. As I mentioned, we're working with a partner on diagnosis to identify people who are at risk of an indication. We're working with partners in treatment eligibility, where patients are on a different drug class, but a combination of a self-management tool for patients along with clinical decision support for HCPs allows for HCPs to drive more confidence to up-titrate patients onto a higher class of therapies. We're focusing, especially with injectables and med device delivery agents, companion or combination digital therapeutics that work to drive therapy initiation so that we're able to get patients on the right treatment therapy because infrastructure on the ground doesn't allow for it. We work a lot with our partners where a drug is already FDA approved and at different stages of launch on market share. How can we drive better market share because we focus on unmet patient needs and unmet clinical needs, which the drug was never designed to solve for, but still matter for that patient population or that therapeutic area. And we use our digital medicine solutions to enable that so that HCPs are more confident to prescribe this brand, this solution over others. Of course, a lot of our work takes us through medication adherence, where we've shown comprehensively how we're able to drive an augmentation to medication adherence across the board, across several of our partnerships. And in some cases, how can we drive net price up, where if the brand is to be bought by a patient out of pocket, or the brand has a reimbursement case that needs to be strengthened, we've now been able to demonstrate revenue centers through our software as a medical device solution and take that at scale as well. We've been really fortunate to now work across very specific use cases. As I mentioned, companion or combination software as a medical device solution with our pharma partners, a standalone prescription digital therapeutic, which is undergoing a clinical study, remote patient monitoring, and patient support sometimes. Clinical evidence, positive outcomes have now been showcased across 30 publications, especially in cardiology and diabetes, which were our primary use cases in 2020. And as we get into our rare disease and our women's health portfolio and those data points come live, you will see these clinical publications come online in 2022 as well. An example of a use case, what pharma loves about us is that we're the base platform, but we allow pharma to own the indication-specific IP on top. We co-design, we co-develop. We are the R&D partner for pharma when it comes to software as a medical device solution. And because we've been able to commercialize a few of them already, We have more than 500 HCPs who have prescribed our solution a few times and have trained a few hundred reps in the process. We also help in market access, and we also help in co-commercialization as we take these products to market. We've got a few of these solutions already out. We have a 90-member team, which is largely based in Asia, with some team members based in the EU and the US, just doing this one thing, which is being this partner for pharma as we attempt to be one of the world's largest companies to build active digital ingredients to enable unmet patient needs. So thank you so much for your time. We're raising in A round later this year between 15 to 20 million. We're using that capital largely to be able to grow into Asia and the EU, and then build as we have a regulatory approval in the US, then build into the US as well. So I'll pause here, happy to answer any questions that you may have. Thank you so much for your time.
Thank you. I think we've got a question from Min.
Yeah, thank you for your presentation. Min from KB Investment. I know you mentioned about the cases, but could you elaborate more on our revenue model, like business model, how we generate revenue? And also my other question is that I think it would be helpful and kind of help us to facilitate understanding that, what's the distinguishing, competitive distinguishment compared to other competitors?
Absolutely. So thank you, Min, for that question. Our revenue model is very simple. We have a platform license fee for the duration of our contracts. Our contracts today run with anywhere between 3 years to 7 years with our pharma partners, along with, in some use cases, especially where we're really excited about our partnership, we get into risk sharing. One option is flat license fee for the duration of the partnership, and as more patients come on board, a patient's license fee as well. Option two, where we're really excited, is we take a risk sharing, which means we reduce the platform license fee to the amount per month or per quarter or per year in favor of milestone-based payments and revenue or royalty share-based payments as a product goes live. And that just allows us a higher kicker on the upside. So we de-risk wherever possible. So we obviously are more aggressive and we want to build revenue and royalty share arrangements because that's more lucrative, but pharma partners need to agree. So we've been able to do that in 2 of our use cases. And in some of our use cases, the flat fee. So that's as far as a revenue model is concerned. And as far as, let's say, our superpower, if you will, what sets us apart from competition, to be honest, it's about the fact that we're able to solve for this at scale. Because of our platform, it used to take us 2.5 years to be able to build out a digital therapeutic and take it to market. We've now been able to do this in 9 months, and that too in the EU. From a cost to market, it used to take us about, and takes our competitors to do this at between 2 to 5 million ARR a year. We're able to do this at more like 250 to 500,000 a year, which sort of sets us apart. Third is because we've been able to do this across several indications, we're able to solve for unmet needs across the comorbidity spectrum, versus a lot of the other sort of competition out there, because they're indication specific, they're able to only solve for that primary indication, but not able to solve for the comorbidities that come with that indication, which doesn't necessarily solve for the brand share or market share problem. And last but not least, our specialty is in companion, combination, and complementary digital therapeutics. So the world is moving and a lot of action is happening on standalone, and we have one in our pipeline. But what sets us apart, because we're based in this part of the world in Asia, we have the good fortune of being able to work at scale in the real world setting as a complement to the drug. And we feel that there's a massive opportunity to generate a fairly large amount of scale as a complement to the drug. And that's where we've focused our time and effort on.
Great. Okay. I think, is there any other questions? We might have time for one more. All right, well, great. Thank you so much for your presentation. Really enjoyed it.
Thank you so much for having us.
Thank you. All right. Moving on then to our next presentation. We are going to be introducing Oncomix and Dr. Stephen Potts, the co-founder and CEO of Oncomix Therapeutics. Oncomix is using a non-pathogenic oncologic myoxic virus for systemic delivery of multiple armed payloads to drive immune responses.
Thank you so much and appreciate the chance to present. I hope you can hear me okay. I'll just share my screen. The first thing to introduce is actually our virus. This is a myxoma virus. It's a large pox virus. It has not been in the clinic before, but it's been proven in about 30 peer-reviewed publications to be lethal both for cancer and to generate a very strong immune response. My name is Steve Potts. I'm the founding CEO. I've got about 2 decades of experience in oncology drug development. Most recently with Ignita. I've had 3 consecutive exits. My last 3 companies have all had a minimum return to investors of 12x. In a nutshell, we're addressing an unmet need in immuno-oncology. There's 3 big things you want to be aware of that you really need in this field of oncolytic viruses to generate something that is really optimal for patient care. It needs to be multi-armed, hit multiple things in the immune system at the same time. Not just one thing, but really going after the multiple things to drive immune response. That's multi-arming. Secondly, it needs to be systemically delivered, IV delivered. The current standard of care in this space is intratumoral. You take a needle and you inject into melanoma lesions. To really get this in something that's very broadly used across immuno-oncology, it needs to be IV delivered and multi-dosed. And then finally, a non-human pathogen. And it's actually helpful with COVID now. This is clearer to people. And I ask people the difference between your first and your second vaccine. You know, the first vaccine, you have no pre-existing immunity. The second one, you do. This is a non-human pathogen. It cannot cause clinical infection in humans. There's no pre-existing immunity. So it's a huge advantage that we're using this. Our pipeline is a multi-armed virus going into the clinic in both solid tumors and heme cancers. And that's followed by a second program, completely different arming, that's going after different tumor types. So basically we have 4 programs with 2 viruses, one heme, one solid, another heme, another solid. And we think this is really what we need to build a public company. It's a great team. We actually added up the number of NDAs this team's been involved in. It's 35 successful NDA submissions. So a lot of drug development experience. Our CSO is an antibody drug developer, came out of Pfizer, and then she actually worked with Chuck Baum, our chair. Chuck is world famous as one of the top immuno-oncology and precision medicine leaders. Previously led the Pfizer immuno-oncology program, and he and Leslie actually worked together at Pfizer. So in some ways, we're kind of going back and thinking about the kinds of immune armings we want to use, and there's lots of things we can put together. We recently closed a $50 million Series B led by Lumira and B Capital. And we've had from the beginning Korean investment partners. They've been a wonderful partner for us, and they've been a Series A investor. We're about a year from the clinic, so we're in our IND and GMP enabling studies right now. And then the founder is Grant McFadden. Like I said, about 30 peer-reviewed publications, about 2 decades of work looking at this myxoma virus as an oncolytic agent and a powerful immunostimulatory approach. The reality is in immunotherapy that about 1 out of every 7 cancer patients are actually responding to immunotherapy, but about 40% of all tumors have at least 1 indication. And if you look at something like colorectal cancer, this is a good example. Only 4% of patients actually benefit currently from immunotherapy. In gastroesophageal, almost none. And in heme cancers, it's almost non-existent in myeloma, myelofibrosis. Incidentally, these tumors are the areas we're going after with our lead program, colorectal, gastroesophageal, and then 2 heme cancers. So it's a big unmet need clinically for what we're trying to do. The immune system is really complex. You want to ideally hit it with more than one thing. Currently, you typically do 1 checkpoint inhibitor, and then you'll try 1 other thing. But we want to really kind of take the innate power of this virus and include multi-arming to basically shut down, to really stimulate the immune system in multiple ways. The field of oncolytic virus has been around for a long time. There's a number of companies in China. There's some really interesting Korean companies. There's an FDA approval, and there's also a Chinese approval of oncolytic viruses. But to really drive and have the potential of this, first of all, it needs to be multi-armed. The past has been that you're going to look for a small RNA virus that isn't armed, take it kind of out of nature, and kind of take what nature gives you. That's not enough. What we've learned is that you need to be able to control and program additional warheads into the virus. It needs to be multi-armed. IV delivered. The current largest public company is an intratumoral delivered with a needle into melanoma lesions. And while it's having really good results, it's not something that the patients really want IV, and the clinics want IV, and really, frankly, the pharma companies as well really want IV delivery. That's how checkpoint inhibitors are delivered. It's really how the virus should be delivered. And that's a high bar. And then finally, non-human pathogen, which, as I said earlier, have just a lot of advantages for this. This is a rabbit virus, and it cannot clinically infect humans. Just as a comparator here for some of the other programs in the space. And the thing you want to know is that if you're going to multi-arm, you need about 20 to 30 kb of space. That means you right now the options are either vaccinia, which is similar to the smallpox vaccine, or the herpes virus. And there's a number of companies working in these two areas. We have myxoma all to ourselves. It's the only virus that is multi-armable, IV delivered, and is a non-human pathogen. It's very amenable to multi-dosing. Another thing very interesting, it's actually carried by leukocytes. So it actually transports in the rabbit naturally, kind of piggybacking on white blood cells. Another thing that's interesting is that in the heme area in blood cancers, both vaccinia and herpes don't work in heme. So in multi-arming, we really have this field to ourselves, which is very exciting. There's been a number of papers comparing vaccinia to myxoma, and what we show on the left is that head-to-head studies with myxoma is it's much more immunostimulatory, and on the right, with this myxoma versus vaccinia, it has a much wider dosing window. There's no toxicity like we saw with vaccinia. So some advantages over kind of the more widely used smallpox vaccine or vaccinia programs. So what we're trying to do here is really take the innate immune system, the power of this virus. This has 80 immunomodulatory genes already in the virus, and it's proven to be super strong as an immunostimulatory agent, and then add some additional arming. So we have some armings in here that drive IL-12, that driving T and NK cells. We have armings in here that inhibit EGFR, TGF-beta, and that kind of modify the microenvironment. So we're adding armings to what the virus already does to get this most sustained immune response. One thing we're finding that's very exciting is that the more we engineer it, it doesn't weaken the virus. If you look at like coronavirus as an example, the more it mutates, the weaker it does seem to get. We don't see that in this virus. So actually, we can modify 2, 3, 5, 6 transgenes and have the exact same behavior by the virus. So this is really an optimum, a platform for being very creative. So our lead program has a couple transgenes in it, but you can imagine there's 30 or so, maybe 40 immunomodulatory genes that we could put in here. And if you have up to 6, there's a lot of things we can do with this virus. We show that this is a 5-armed virus. It's got TNF, decorin, IL-12, and then 2 different protein reporters in here. You see a lot of production in the green and the red of those 2 reporters. And what you're seeing is a very strong amount of protein produced. So that's exciting. And then we see that this protein is functional on the right. So we do functional assays on this. So we can produce a lot of virus, multiple proteins, and have them be functional and have it not affect how the virus actually behaves and replicates in tumor cells. Remember, this virus will go in, it'll replicate only in tumor cell and create a sustained immune response in the local tumor environment. This slide shows something that is really important here, which is it answers the question of you want to see an effect by the virus on its own, and then you want to see an effect by the transgenes. So the blue is the vehicle, the green is the myxoma virus, and then in purple is a double-armed virus. And what you see is that here, with the stimulation of T cells, you see that it's really being driven by the armings, in this case, the IL-12 arming. And over here on the right, what you see is that the virus is actually reprogramming the macrophage ratio. So the virus is doing a macrophage ratio reprogramming, and then the armings are doing a T cell reprogramming. So they're working well together. We have a lot of data, both IT and IV. We're very committed to IV. We've shown IV delivery in 4 different mouse models at acceptable, clinical acceptable doses. And by the way, whenever any of these mice that are completely where they're cured from the cancer, when we re-challenge them, you don't see the tumors growing again, which is exactly what you want. We're also seeing that the the virus will accumulate and grow in the tumors with IV. This is spectacular. You don't see that. It's really been very difficult for the industry to see this kind of data. So you actually see with an IV delivery that the virus is growing in the tumor, but not in the liver or the spleen or in bone marrow. Here also you see more virus accumulations, exactly the kind of data you want to see, to really convince yourself that the virus is going to grow locally only in the tumors. We have about 10 peer-reviewed publications that show very strong data that show that the virus is lethal for multiple myeloma and another actual blood cancer too. So in this case, you see that 100% survival of mice that had multiple myeloma, and you see a very strong death rate of those that did not have the virus exposed. So it's a powerful agent both in heme and in solid cancers. We've got to the point where we've locked our development candidate about a year ago. We're in the middle of IND. We have a pre-IND meeting in the middle of that, and then we'll have our first IND Q1 of next year. So about a year from being in dose in humans. Manufacturing of US-based manufacturer. We have all the things you need. We have a serum-free cell line. We're using a scalable process and a really strong manufacturing partner. So we're seeing, very good CMC, and the kind of data we're seeing at the CMC is very similar to what we see in our research site. So regardless of how many transgenes we put in, we still see very good yields. The lead, if you think of this kind of as betting in some ways, the lead program is with 3 approaches we're taking here. Monotherapy in solid tumors, monotherapy intratumorally delivered in solid tumors, and then also a heme program. And this, again, is very unique to have both a heme and a solid tumor program going concurrently in the oncolytic virus sector. This is going to allow us to really demonstrate programmable precision medicine with our solid tumor IT program. And we can look at a cohort that's likely be better responsive. And then we can look at IV 2 different ways, both in solid tumors and in heme. And again, on the pipeline, so there's 2 viruses. OM1 is our lead virus. OM4 is our second virus. These are completely different armings. So that's important because it allows us to partner the second program. So we are interested in potential partner opportunities with the second program. We're going after some very interesting tumor areas with high unmet need. And then we have other research programs in discovery stage right now. So with that, I'm going to try to catch us all up on time a bit. Just the thing you need to know about this space is that the oncolytic viruses are very attractive as a way to increase the immune responses. Remember, currently immunotherapy, 1 out of every 7 patients is currently responding. So there's a huge opportunity if we can get the response rates to improve. To do that, we need 3 things: multi-arming, IV delivery, with a non-human virus. And it lets us be very creative with the kinds of targets we can approach that might have had prior systemic tolerability issues. Things like IL-2, IL-12, TNF, IL-15, there's a lot of them, bispecifics. We can be very creative with the kinds of targets we can put in as we demonstrate that we can IV deliver and have local delivery of this virus to the tumors. We're fully funded next 2 years, and we're in good shape. So with that, I'll stop.
Great, Dr. Potts. I know there's a lot of questions for you, so maybe we'll start with Min.
Yeah, Min from KB Investment. So my question is, so as I understand, we don't humans probably have no pre-existing immunity, which makes sense for a single dose because before that the immune system comes in, that makes sense. But how is it possible if we, to have multiple dosing with this when immunity kicks in?
Yeah, so with any virus, you're not going to use this dosing forever. You want to dose for a certain period and then create a sustained immune response, almost like a vaccine response. We think the optimum amount is about 6 to 8 weeks. And so our clinical regimen is basically weekly for 6 to 8 weeks. And we think that's about the window for, after those 8 weeks, you will then have some acquired immunity. And so you'll see some tapering off. We won't know for sure. We actually see that the immunogenicity profile is very attractive. But again, we're just basically planning on 6 to 8 weeks with weekly dosing. And so you will see an increase in immunogenicity over time over that period. Great question.
Let's move on to Erin.
Hi, this is Erin from Intervest. And then I have two questions. And then one is respect to the delivery method. Comex is trying to deliver by IV, and then I understand that the myxoma virus is one of the pox virus family, but it has like tumor tropism. But do you believe it is good enough for in human delivery as well? And then the second one is that it is like hot field for checkpoint combination therapy for the cancer vaccine and the checkpoint inhibitor. Do you have any plan for the combination therapy? And then if so, which checkpoint inhibitor are you thinking about?
Great questions. The first one, we've tested about 30 or 40 different cell lines, and we're always looking for, will the virus infect normal cell lines versus tumor cell lines? What we've consistently seen is that the virus replicates in tumor cells, and it pretty much just tapers off and is killed quickly in human lines. And actually, the virus is widespread in the environment. So it's had an opportunity for millions of years to try to infect beyond rabbits, and it hasn't been successful. It was used in multiple countries in rabbit pest control, and it's actually so specific that in Australia, it could infect Western rabbits that were not native, but it leaves the Australian hare alone. So it's a very, very specific tropism, and it widely infects tumor cells, but it simply just kind of leaves the normal cells alone. So that's been demonstrated in a lot of papers. In terms of, a great question on the combination therapy. We're currently in discussion with several partners. There's a lot of interest from the large global players in immunotherapies. You probably can imagine PD-L1 type programs and also PD-1 programs. That being said, we would welcome opportunity from any also from Asian pharmaceutical companies developing checkpoint inhibitors. And I think this is a chance to really differentiate because ultimately everyone's going to have a checkpoint inhibitor. So this is a way, the reason why the partnering opportunity is so attractive financially for these pharma companies is that if you can see better responses, it's a way to have a huge differentiation for your checkpoint inhibitor program from other things going on. So, and our discussions are currently underway. Typically, you want to have a checkpoint inhibitor partnership about 1 year prior to, which is we're about 6 months from needing to kind of sign a deal there.
Great. I think we have one more question from Sumin.
Okay, this is Sumin from Partners. Many part of my question was answered from via previous questions, but just to make sure, so tumor tropism of the oncolytic virus has been shown in vitro many times, but it doesn't really repeat in human. So to avoid those problems, and they sometimes make it replication incompetent or only replicate in the tumor cell. Do you do any those type of manipulation? It doesn't look like your virus look like replication competent, right?
Very replication competent, and actually any adenoviruses are typically not replication competent. We do some engineering to make it even more powerful as a cancer killing and immune killing agent, some proprietary things that we've learned, but we actually don't need to do any attenuation from a health standpoint to make it less worrisome from the clinical. It's a big advantage. It just simply isn't even close to being able to clinically infect humans.
Okay. Do you select any specific substrain of myxoma virus?
So our founder spent his whole career, 30 years with myxoma virus. So we've used we have built this on a very specific kind of optimized approach over the years that there's about 10 patents around this. And then on top of that, we do the arming. So it's not something you can just go to the biobank and start with the myxoma virus. You would not want to do that.
Okay.
It's again, it would be very difficult for anybody else to play in the myxoma space. We have intellectual property wrapped up globally, and including all the major Asia country players. Great question.
Okay. Thank you.
Thank you so much for your presentation, Dr. Potts.
Thank you. Great forum. Really appreciate it.
Thank you. All right, we're going to move on to the next presentation. The next presentation has been pre-recorded due to the timing issues, and it's with Myrexus. The presentation is by Dr. Lihan Zhao, co-founder and CEO. Myrexus is a microRNA-based early detection test and comparison diagnostic for cancer and other diseases.
Good afternoon. My name is Li Han. I co-founded and have served as the CEO of Meraxis, a Singapore-headquartered molecular diagnostic company with physical presence in the United States, China, Japan, and Southeast Asia, with more than 300 staff globally. Even though we couldn't meet in person for the second time in a row, it is my pleasure today to present our technology, clinical pipeline, and commercial progress in virtual. We're fundraising and we'll be very happy to arrange one-on-one meetings to further the discussion. You'll find our contact details on the last 2 slides. Our disclaimers. We'll be most excited about 2 opportunities. First, to develop novel blood-based cancer early detection tests powered by our unique microRNA technology. We're focused on both high-incidence cancers of global impact, such as lung, breast, and colorectal cancers, but also for cancers causing significant healthcare burden in Asia, such as stomach and liver cancers. Secondly, we're excited to be building the leading precision genomics platform for the underserved but growing Southeast Asian market, providing diagnostic solutions across the full cancer care continuum for cancer early detection to precision oncology, as well as in the emerging field of population health. The journey of Meraxis is a 2-decade-long scientific pursuit. The university lab, which the founding team originated from, was the first academic PCR lab in Southeast Asia. We were collaborating with NIH and Roche Diagnostics in early 2000 to develop dengue RNA tests, which is a big problem in our part of the world. We were also among the global pioneers in microRNA science and technology, entering the field in 2003, just 2 years after human microRNA was found. Over the years, we have systematically matured our high-performance detection technology and pioneered blood-based microRNA cancer diagnostic development, first in stomach cancer, and then in multiple cancers, as well as cardiovascular and pulmonary diseases. We conduct research and clinical studies globally with leading clinical and academic institutions. Meraxis was officially founded in Singapore in 2014 and has since expanded our presence to the United States, China, Japan, supported by 3 rounds of venture financing by both the American and Asian healthcare specialists. Today, we're focused in accelerating the translation of microRNA science into clinical diagnostic tests, powered by our proprietary M-Smart qPCR assay chemistry that's been patented globally. GastroClear, a blood microRNA-based PCR test, is a first-in-class molecular screening test for stomach cancer, and it's among the very few molecular screening tests that has been approved globally, which has undergone prospective clinical trial of at least 5,000 patients. Our decade-long experience in discovering microRNA biomarker and developing clinical tests are now enabling us to accelerate the development of a pipeline single cancer, as well as a multi-cancer screening test powered by our microRNA-centric multi-omics approach. Over the last 7 years, we have also built significant commercial presence in major markets, as well as developing countries in Southeast Asia, generating more than 50 million in revenue in 2021. We've been most excited by the unique biology of microRNA, where these molecules are actively and selectively secreted by cells as means of intercellular communication. microRNA circulate in the body fluid at an abundance which is much higher relatively to that of circulating tumor DNA. This has translated into a much higher sensitivity using circulating microRNA biomarkers in detecting early disease cancer as compared to ctDNA, while ctDNA holds an advantage in specificity. There's been a strong growth in microRNA publications and clinical evidence with close to 120,000 publications since the discovery of human microRNA and more than 1,000 registered clinical studies in clinicaltrial.gov. Being the smallest genetic material found in human, animal, and plants, it is extremely difficult and challenging to detect microRNA robustly. Our patent and smart qPCR assay chemistry allow us to specifically bound the target microRNA, converting those microRNA into cDNA, and robustly amplify and detect those through a PCR approach. Comparing to the probe-based approach by multinational companies, the 3' approach which Meraxas has developed and patented allow us to achieve the level of analytical sensitivity, specificity, and robustness that warrants clinical translation of these novel science. Over the years, in addition to the proprietary qPCR assay platform, we have conducted retrospective and prospective discovery cohorts, including more than 50,000 clinical specimens, identifying a series of novel disease biomarkers. And by combining the technology and the novel biomarkers, we've then developed IVD test kits and undergone clinical trials. So the combination of the technology, the biomarker, and the test algorithm IP, plus the trial data, has built a significant high entry barrier. GastroClear is our first clinical product based on our microRNA technology. It is developed to address the screening needs of stomach cancer, which is the fourth deadliest cancer worldwide, with more than 1 million new cases and close to 770,000 lives lost every year. Early diagnosis and intervention of stomach cancer results in at least 3 times higher survival rate and significant treatment cost saving, as demonstrated by Japan and Korea, the only 2 countries with nationwide stomach cancer screening. However, the only means of stomach cancer early detection today, as demonstrated in those 2 countries, is through gastroendoscope, which really isn't suitable for mass screening. So we developed GastroClear as a blood test that will address the accessibility and affordability of stomach cancer screening. Through 7 years development journey and a prospective clinical trial, we have demonstrated the ability of such a blood test in detecting a significant amount of early stage cancers, and GastroClear has been approved for screening needs in asymptomatic at-risk population. By developing the test on a PCR platform, it also enables accessibility and fast turnaround with as fast as 4 hours of sample to result time. And with a global buildup in PCR capabilities, now we can bring the cost of running such a test down significantly, which will enable mass screening. The goal of GastroClear in moderate to high incidence country is to use such blood tests as the first line screening tool to screen the at-risk population, identifying the high-risk population, which warrants further endoscopic intervention to identify and treat early stage cancer. The development, analytical, and clinical validation of GastroClear was benchmarked against Cologuard, the first and the most successful molecular cancer screening test. Through the 7-year development and 3-year prospective clinical trial enrolling more than 5,000 individuals, GastroClear has been proven to outperform all existing blood-based biomarkers that's been tested for stomach cancer, demonstrating at least a 20% advantage in AUC from the closest competition, whether it's H. pylori antibody, pepsinogen, CEA, or CA19-9. We're most excited about the sensitivity of the test, which allows us to detect close to 90% of stage 1 gastric cancer and 60% of stage 0 gastric cancer or high-grade dysplasia, including 75% of those lesions less than 1 centimeter. The positive predictive value and the negative predictive value of GastroClear is similar to that of the existing cancer screening tests, including Cologuard for colorectal cancer, LDCT for lung cancer, and mammogram for breast cancer. Through this journey, we have taken the best-in-class technology to commercialization by building substantial clinical evidence, manufacturing know-hows, regulatory approval, health economics, as well as educating the market. With a successful Singapore trial and approval, we have since launched registration studies in Japan and China and would expect approval in the next 1 to 2 years. In addition to GastroClear, we have also built in the last 10 years a strong and maturing pipeline of single cancer microRNA tests, as well as a multi-cancer screening test powered by a unique first-in-kind approach combining microRNA and methylation. Our clinical progress is published in leading scientific and medical journals and presented at global conferences. Leveraging the R&D and commercial capabilities developed for microRNA-based cancer early detection tests, we have since expanded our development and clinical test menu to cover therapy selection, prognosis in the underserved Southeast Asian precision oncology market, and have started building tests for the emerging field of population genomics. The same capability also allowed us to contribute meaningfully when the COVID-19 pandemic hit last year. We worked with the Singapore government to deploy Singapore's first approved COVID-19 test, not only in Southeast Asia, but now in more than 40 countries worldwide. We commercialize our innovation through a synergistic approach of selling IVD test kits and providing clinical testing services either through the self-owned lab or partner labs. We are actively expanding our oncology testing markets in Asia, in Southeast Asia, Japan, China, and heading towards the United States in the longer term. We have built long-term partnership with key academic clinical institutions to effect guideline setting and demonstrate clinical utility. Our IVD test kits enables in-house testing for faster turnaround time and minimizes logistics and reimbursement challenges as faced by the academic clinical institutions. Our medical affairs and commercial team are also active in driving adoption in private screening clinics and among family physicians. Instead of selling test kits, we provide clinical testing services for these private clinics that often do not have in-house testing capabilities. What we are particularly excited is our ongoing work with payers to utilize novel clinical diagnostic tests to help payers manage the growing cancer treatment claims while reducing premium for policyholders. We are experimenting novel cancer prevention approach with payers in Singapore and Japan. COVID-19 has increased PCR or molecular testing capabilities globally, especially in developing markets such as Southeast Asia. Through our pandemic response effort, Meraxus has built significant customer base and lab partners in majority of the Southeast Asian markets. The branding and the trust built through deploying more than 8 million COVID-19 PCR tests in the region is now driving the adoption of our oncology solutions. We expect more and more PCR labs in the region to switch focus from COVID testing to molecular tests focused on longer-term unmet needs oncology, thus adopting our novel cancer diagnostic tests. In the past 7 years, we have grown Meraxus from an R&D-focused life science firm to a regional powerhouse in molecular diagnostic tests, capturing the growth in oncology molecular tests in Asia. Powered by our unique technology platform and the maturing commercial capabilities, we're confident to grow significantly in the coming years by commercialization of our pipeline of cancer early detection tests in global markets with a focus in Southeast Asia, China, Japan, and US, while building our leading position in precision oncology in the underserved Southeast Asia. We've been busy since the completion of Series C financing in 2021. Just last year alone, we have launched 3 prospective studies with significant public co-funding in Asia, with a target enrollment of 17,000 individuals across liver, colorectal, and a multi-cancer screening test development. 2022 will see the launch of our second microRNA test for lung cancer screening as an LDT in Asia, allowing us to start gather real-world evidence to guide the design of registration trials. We're also on schedule for the registration of GastroClear in China and Japan, where China trials officially launched just last week, enrolling 12,000 individuals. Towards the second half of 2022 and early 2023, we're expected to have first readouts of our 2 single cancer tests and the exciting multi-omics multi-cancer test project. The 300-strong staff in Meraxus across the globe is led by an international leadership team and advised by the best scientific minds in the field of microRNA and clinical diagnostics. Praxis has also been recognized both regionally and internationally for science, technology, and entrepreneurship. We're excited with the clinical impact that our technology platform can deliver and would very much welcome like-minded investors to join us in this journey. Please connect with us either through emails or through the JP Morgan conferences platform for one-on-one meetings. We have set aside most of the slots in January 18th to 20th. Thank you for your attention. Stay safe. We'll talk soon.
Great, thank you so much for that presentation. We encourage anybody with questions to reach out directly to Dr. Zhou to answer any of your questions. And we'll move on now to our last presentation. For the same exact reason, this presentation is pre-recorded as well. The presentation is by the President and CEO of PDC Line Pharma, Eric Halioua. And PDC Line Pharma is developing off-the-shelf cancer immunotherapies based on its PDC line.
Good morning. My name is Eric Halioua, and I'm the CEO of PDC Line Pharma. I'm going to present you over the next 12 minutes the activity of our company, which is developing a new class of cancer vaccine based on an off-the-shelf antigen-presenting cell line. In a nutshell, the company is a spin-off from the French Blood Bank. We are today a team of 27 people, and in terms of funding, we raised close to 52 million euros, a bit more than 31 in equity, and the rest in non-dilutive money. The technology we are developing is coming from the French Blood Bank, which is a very large organization in France that has the mission to collect blood, but that have R&D associated lab where the scientific founder, Joël Puma, was working. And he has been able to identify in the blood of leukemia patients a very rare leukemia called plasmacytoid dendritic leukemia. In fact, in the blood, you have the dendritic cells, which are the cells, the professional cells to present antigens to the immune system. And the big part are the myeloid, and the tiny fractions are the plasmacytoid. And the leukemia has been affecting this tiny part of dendritic cells. And these cells have very interesting biological property. And based on the blood of this patient, he has developed a master cell bank, GMP approved for clinical use, that we are still using today as a vaccinal platform. So our proprietary PDC line has 3 key advantages. First of it, it's an immortalized cell line, so it's extremely easy to expand in culture. It's a plasmacytoid dendritic cell, so the origin of the cell provides very interesting potent antigen presenting capabilities. And I will show you some data. And it is a versatile platform. In fact, we can apply by just adjusting the peptide we are loading to the cell line, to the PDC cell line, to any kind of solid or liquid tumor. We are a clinical stage company. We have already completed a phase 1b academic study in melanoma, and we have ongoing a phase 1/2 in non-small cell lung cancer. The pipeline of the company is currently constituted of 3 key components. First, the PDC male, that was the first vaccine candidate we have developed and where a first clinical trial has been completed. The result has been published, but this study is not active anymore. We have a second candidate, which is called PDC lung, with 6 shared tumor antigens, which is ongoing on non-small cell lung cancer. And we have at preclinical stage another candidate on neoantigens. Important to mention that we have already validated the value of our platform thanks to the deal we signed early 2019 with LG Chem to develop the PDC lung cancer vaccine in Asia. In fact, LG Chem is a well-known organization in Asia, and they have a life science department which is definitely growing. We granted exclusive license in South Korea and exclusive options in other Asia countries for the development and commercialization of PDC lung cancer. And as you can see here, the total deal value is more than $123 million, plus significant tiered royalties on net sales. The team of the company is definitely the management team, one of the key assets. I am myself co-founder of several biotech companies. I've been involved in IPOs, M&A. I raised more than 150 million euros in my career. We have Shanna De Bruyne, as you can see on the bottom, which is a very experienced medical director coming from GSK Vaccine. She led the cancer vaccine, MAGE vaccine within GSK, the phase 3 clinical trials. Claude Dédry, the VP of Manufacturing and QC, has a long experience in cell therapy and is coming from GSK Vaccine as well. Joël Plumat, the scientific founder, joined full-time the company. Laurent Lévy, the COO, CFO, as well, is full-time within the organization with large experience in oncology. The company made significant achievements over the last 24 months, and you see here the list of its achievements. In December 2019, we closed a 20 million B round of financing led by the Korean VC, Korea Investment Partner. It came, in fact, in consortium with 2 other South Korean investors. In February 2020, we administered a first patients, non-small cell lung cancer patients with the vaccine PDC-Lung01. We announced in April the publication of the international patent applications of the modified PDC line. The president of the board, we have a very competent president of the board, Monder Majoubi, which is the former executive VP of oncology of AstraZeneca Genentech. He's an oncologist that is now the president. He arrived in June. In September, we announced the administration of the first patient with high dose. We get the authorizations to start the clinical trial in Germany in November 2020. In December, we announced that we get 100% compliance of all the batches produced this year in 2020. We made another publications in February 2021, opened the last cohort of the clinical trial for PDC-Lung, which is the high dose added to checkpoint inhibitor in September. And more recently, we announced the closing of a B2, a round of financing of 17.5 million euros, led again by Korea Investment Partner, with arrival again of 3 new Korean investors. So today we have 6 South Korean investors within the shareholder of the company. So let's pass now to the presentation of the technology. So how it works. So the cell line I presented earlier, coming from the blood of leukemia patients, is stored in liquid nitrogens. What we have to do when we want to do a campaign, manufacturing campaign, is just to seed a single-use bioreactor with a bag of the working cell bank, and the cells grow in suspensions in a synthetic medium without any carb serum of interleukin or cytokine. And after 9 to 10 days of culture, we have more than 20 billions of cells. Then at the harvest, we just have to split the cells according to the number of peptides we want to stick on the HLA of the cell line. Each peptide is representative of a tumor antigen. So we select the tumor antigens according to the cancer we want to target. We had 4 for melanoma, but we have 7 for the lung. Then we, after the loading, we pull the cell, irradiate to stop the proliferations. Then we put in a specific container. It's the technology of aseptic technology, where the cells are preserved with the cryoprotectants in liquid nitrogens. And when we just have, like for the cancer vaccine, to ship the cell line in a minus 80 degrees fridge in the hospitals. And when the patient is ready, we just have to take the vial, thaw the cells, and with a syringe, take the cell with the benium and then fuse to the patient. So it's extremely simple process that has been successfully already implemented in the current clinical trial. So the second key advantages of our cell line is the potency. So we did several benchmark studies to assess the capacity of the cell line to expand in vitro CD8 T cells specific of antigens. And here we have selected melanate. And you see here the capacity of the cell line to expand the CD8 T cells compared to classical approach, which are the conventional DC, autologous or allogeneic. And as you can see here, measured by FACS dextromer, we are 20 to 200-fold more potent than the classical approach. This study has been published. We did as well study with neoantigens. We have selected 2 neoantigens published in the literature, identified in the blood of lung cancer patients or melanoma patients, where the identification of the mutation is clearly established, as you can see in red, and loaded the cell line with each of these neoantigens, ME1 and ACAP13, and with the blood of 3 different healthy donors. So it's purified CD8 T cells of the 3 healthy donors. And as you can see here, after 21 weeks and 21 days, and 2 weeks of culture, as you can see in red and purple, we are definitely able to expand the CD8 T cells specifics of each of these antigens. And we reach a fold increase of 180. And that is extremely high, knowing that we have with healthy donor all the precursor of the CD8 T cells of the neoepitope that are naive, and the frequency is extremely low. It's 1 out of 1 millions of the circulating CD8 T cells. We did as well study to demonstrate that the vaccine in combinations with the checkpoint inhibitor can have a synergetic effect. And for that, we have collected the blood of melanoma patients. Melanoma is a very hot tumor with a very high tumor-induced immunosuppressions. And we have loaded the cell line with, again, melanate and put culture with the PBMC of these melanoma patients and measure after 1 week the expansion by FACS dextromer of the CD8 T cells specific of the melanate. And as you can see here, with the vaccine alone, we have 15-fold increase. With the checkpoint inhibitor alone, we have 2-fold increase compared to baseline, so it's not statistically significant. But when we combine both the vaccine and the checkpoint inhibitor is when we observe this very nice synergetic effect with a 30-fold fold increase, which show clearly the synergetic effect with the vaccine. In terms of IP, we have 3 key pillars in our IP strategy. First of it, we have the cell line, which is unique worldwide. It's coming from 1 patient. It's a master cell bank, which is clinical grade, and we have the exclusive license from the French Blood Bank. We have the patent. 2 patents has been already granted, and we filed a new patent in 2018 and the genetic optimization of the PDC line. And the third level is the know-how and data related to the manufacturing process and QC, in vitro bioassay, immunomonitoring assay, and all the preclinical data generated so far. Let's pass now quickly to the non-small cell lung cancer current trials, which is ongoing. So this is, you see here in this slide, the 6 tumor antigens we have selected, MAGE-A4, MAGE-A3, MUC1, New York ESO1, Survivin, and Multimage. And this is the antigens, the peptides that are loaded to the cell line to make the vaccine. And as you can see here, in the blood of healthy donor, for all of these antigens, we are able to have a very nice CD8 specific, antigen specific, CD8 specific expansions. That is done after 2 weeks of culture. The design of the current clinical trial is presented briefly here. We are working on 64 patients. The main objective is to assess the safety, the immunological activity, and the clinical response of the lung cancer vaccine in association with checkpoint inhibitor. And we have 2 doses of vaccine we are testing, low dose and high dose. First in monotherapy in adjuvant setting, 6 patients for the low dose, then we passed to 10 patients to the high dose. And after we have a second population, which is the first line metastatic patients in association with checkpoint inhibitor with a tumor having a PD-L1 expression above 50%. And here for the low dose, we have 6 patients. And for the high dose, we have 42 patients. So we have already completed the 3 first cohorts, and we are now ongoing on the last cohort of patients, the 42 patients. Here, we are infusing 6 times the vaccines weekly, once a week. To finalize my presentations, the funding strategy of the company, we raised, as I said, 20 million euros beginning of 2020. We did a B2 round of financing end of last year of 17.5 million euros to completely finance the current clinical trials. The objective is to launch a second clinical trials that would be a phase 2/3 that would be in preparation in 2024. And that will be a multi-regional study that we'll do in partnership for Asia with LG Chem. We will require a new funding, and we can make whatever around C or an IPO. IPO in Asia is one of the options. And with the money we will collect, we will set up in US and Asia, and we will carry on the randomized study and develop new clinical trials for new indications. And on that, I would like to thank you for your attention. And
Great, thank you so much. And again, if you have questions for Eric and for PDC Line Pharma, please contact Eric directly. And I think that wraps up our presentations. On behalf of BioKorea, BioCentury, and Sidley Austin, I'd like to thank all the presenters for their really interesting and great and dynamic presentations, and also thank all the investors for their wonderful questions and for their participation today. And I'm going to hand it back off to Jury for closing remarks.
Thank you, Lauren, for being our moderator today for the 8 excellent company pitchings. Thank you for watching, everyone. If you have any questions or suggestions, we're really happy to receive those questions through email. We hope to see you face-to-face at our offline event next year at the actual JP Morgan Healthcare Conference site. So stay safe, stay healthy at this time of pandemic. See you next year at our 3rd Global IR at JPM. Thanks again for watching BioTV.